Master!
Ah, what is it?
Warn your son. Warn your son. Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son or come to my house?
Oh, Ejapia. Ah, what's wrong if Kofi plays with your son? Are we not from the same clan?
Hey, don't you know there is a case on you and your husband?
Me and my husband?
Yes.
Me and my husband?
Yes.
Very good. My husband is in.
Did you say case? All right. We'll see who that case is on.
Deal with him. Case him. Deal with him. But shipping. Come on. Wow.
Jonathan, we can't find Amma.
Where is she?
I don't know, we've looked everywhere.
What? Yes. In that critical condition? Oh God, this is unbelievable. We have to find her. We have to find her at all costs. I'll handle it. My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? Well, you know the people who did all the antenatal tests. How can you tell us our child is deaf? We'll solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. In your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. I think that is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Chikere. Sumanga. Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 canas.
Your Highness, can we pay in cash?
Oh, the gods can take cash. Even mobile money. Chikere, pala wensu.
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali.
He said she was going to get better. when we get home. Now, her condition is worsening. Please help us, please.
This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I am going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. Jesus' name. Oh, Father. Jesus. Jesus' name.
Koyo, she's not getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong. We now have a better understanding of what genetics is and its implications on deafness.
How can we find doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Oh, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, Doctor. Then I'll quickly arrange for the meeting.
Thank you. Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana wants to meet everybody at the village square. The great Asparrow has spoken. Ago! People of Asempa, Nana and the council request your presence. Nana wants to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son Kojo and daughter Agnes are here with some medical officers from Legon to educate us on the subject of deafness and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Look on this chart. It will help you understand it better. Please. Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribose nucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code. Genes variations are like spelling errors, where, for example, there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything the parents could have caused or prevented in any way. They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have 2 copies. One copy comes from the mother and the other copy from the father. In an autosomal recessive condition, an individual needs both copies of genes to be faulty for a condition to develop. The parents of a child usually carry one faulty gene of the gene pair. They are healthy carriers. It is estimated that every individual is a healthy carrier of several faulty genes, but a recessive condition only occurs when both individuals in a couple happen to carry the same faulty genes. When healthy carriers have children, there are 3 possible outcomes. 1. The child inherits both faulty gene copies and has the condition. 2. The child inherits only one faulty gene copy and is a healthy carrier like his or her parent. 3. The child inherits both working copies of the gene. So each time healthy carriers have children, there is a 1 in 4, 25% chance that their children could inherit both faulty gene copies and develop the condition. When an individual with a recessive condition have children, their children will usually all be healthy carriers of the condition. Their children could develop the condition if their partner happens to be a carrier of the faulty gene, but this could be a rare occurrence. Brothers and sisters of a healthy carrier could also be healthy carriers themselves.
So as you can see,
all right. Good morning. I hope everybody enjoyed yesterday, and I hope everybody had a really good night's sleep. Before we get started, may I ask if those at the back could move forward a little bit? Please indulge me. You know, you want people to know who came on time and who came late. So you move forward, and then that way we can tell. Prof. Kwe, you are not included, so all right. So I think we're about almost halfway through. I want to believe that yesterday was quite a good day. We had a fantastic keynote by Emma Wall. Yesterday was quite a good day. We had a fantastic keynote by Emma Wall, which basically and I think it was reiterated a little bit during the presentations that the more we know we need to know, right? Particularly about the immune system pertaining to viral infections and things like that. And we had a number of very good talks, and I believe we're going to follow that up today. There's no opening session today, so it's just serious business. And we'll have a fantastic keynote to continue the trend. And halfway through the day, we'll have also a panel discussion, which will be moderated by me. And that's really trying to sort of put into granularity the whole point of what we do, why we do what we do, to get from basic science to public health impact. And that will come after lunch. Today also, we'll have the start of our poster sessions. Those who have posters, I hope if you haven't verified, check at the coffee break that your poster is up. And you will receive a link halfway through or sometime during the day. We're trying to really be very democratic about the scoring process. So everybody's going to get a link to score posters. And I entreat, if you can, please do. There's a modification on the link. Those who got the link to score the orals yesterday, I realized that you were forced to score everything, and that's going to be modified. So those who didn't score for that reason yesterday, if you can retrospectively remember the scores, I would appreciate it very much. So I think we're about 5 minutes behind schedule, but I think as yesterday demonstrated, falling behind schedule is not that big of a deal because we'll catch up. And it's my distinct pleasure to introduce our keynote for today, Professor George Che. And I don't do very well with reading the prompts and things like that, so I'll go with just speaking naturally. I asked them to project his profile. He's an associate professor of virology. He's an associate professor of virology, but he also runs a group in the Washington University, where he's also an associate professor of, I believe, molecular medicine. And so the reason we asked him to give this keynote is he has built a very powerful team here at the University of Ghana. He's also the scientific director at the University of Ghana Medical Center, and where he's really trying to bridge the gap between clinician scientists and basic scientists. And he's built a team focused on HIV cure research and expanded that to a network across Ghana. Those who don't know about virology in Ghana particularly is that the business here in research is malaria. And I remember when I first came and I was talking to Gordon, and he was like, well, the prevalence rate is 1.3% at the time, and yeah, people don't really think about it that much. And now we went for a retreat for a project, and I see that Prof. Che brought people from across the country, and he's built a network really to take advantage, one, of the low prevalence, but also to there's something uniquely different about West African epidemic, and he's brought all the key players together, and I think he's going to tell you a little bit about his journey, how he manages to be in Ghana and in the US. Maybe, I don't know if he's going to talk a little bit about the US, but we asked him to talk a little bit about the path and the future, and I think it'll be a fantastic talk. So I'm going to stop the rambling now, and I would like to call Professor George Boateng Che to give the keynote of the day.
All right. Thank you very much, Peter, for the invitation. And I want to thank WACCBIP, and all the leaders, Gordon and everybody else, for doing me this honor to be a keynote at this meeting. So we are very grateful, me and my group. So today I will talk about what we've been doing here at the University of Ghana when it comes to HIV cure. So let me just go right into it. So HIV is still a global threat. Over 40 million people are still living with the virus. And since the pandemic started in the 1980s, it has killed close to 40 million people. So that is like the population of Ghana and Burkina Faso, combined. Imagine all of those people dead, right? So it is a big deal. Of course, in Ghana, our prevalence is much lower. I have about 330 persons living with the virus. But that does not minimize it in any way. When it comes to HIV, I think there's no disease that has gotten so much resources and research probably than HIV. And that has paid off. I mean, HIV is a testament that if you put in the resources, you are going to get results. As at now, we have all of these medications are available for HIV. And we have 20 fixed dose combinations. So like those are like doses of several medications combined to treat the patient, so that patients don't have to take too many pills like they used to in the early 90s. So this is a huge advance, and those of us who are infectious disease physicians are very grateful, and we've seen the transformation from 14 pills to 1 pill a day, and transformation from a lot of side effects to virtually no side effect, and is really, really satisfying to treat your patients. That said, the medication that we have at the moment does not or do not cure the virus. So as an ID doc, I like to cure people, right? So like you come to the clinic, you have pneumonia, I give you the antibiotic, you are cured, and I'm happy, right? But when it comes to HIV, it's not like that. The medications are pretty good at suppressing the virus. So if I get a patient and I give him the current cocktail, which is usually 1 pill once a day, they are able to suppress the virus within weeks of treatment to where it gets to undetectable. Undetectable means that by conventional PCR, when we do it, it will say that it is below detection or less than 20 copies per ml in the blood. Of course, that does not mean the virus is not there. It is still hiding somewhere in the body, but this enables the immune system to be reconstituted, and the patient is able to live a relatively normal life. However, if we are to stop the medication for any reason, then the virus comes back. Usually within a few weeks, they can go back to where it was before. So that is the challenge with HIV, is that the virus does not go away. We are able to control it. In other words, that become like a chronic disease, not like the way you control your blood pressure or in any of those conditions. So why is that, or what is happening here? There are a number of problems with this virus that is preventing a cure. The first is the life cycle, right? So HIV enters the cell, and then it makes its way all the way to the person's DNA, and then it integrates into the person's DNA. So it becomes part of the genome. And it doesn't just do this in any cell. It does this in the T cells, right? So if you have your T cells from your immunology class, you know that eventually you're going to have resting T cells. And these resting T cells are going to be carrying the virus. And so the current ART is able to take out the virus when there is active replication of the virus. However, when the T cell is quote-unquote resting, then it is impossible for the ART to reach it. So, as you can see here, there's a slew of places where the virus tends to hide. So there are cell types, there are all kinds of memory T cells, and even monocytes and macrophages. Majority of what we call the HIV reservoir are replication defective. What that means is that they don't have the full complement of the viral genome to make a new virus, about 80%, but the rest are replication competent, and those are the ones that we are worried about. But even the replication defective are able to make viral proteins, right? And when they make viral proteins, they lead to what we call immune activation, which leads to early development of non-communicable diseases like hypertension and cardiovascular disease. So just having even a non-replication competent virus is a problem for the person with HIV. This study is old, but I love it so much because nothing has changed in 20 years when it comes to how the virus behaves. So if you take a number of patients that were followed for 200 weeks and looking at the viral DNA in PBMC, which can be considered as a viral reservoir, and you can see that if you take a particular person, there is virtually no change, right? So the viral reservoir, that is a virus that is within the resting T cells, remains virtually the same over a long period of time. And so the modeling has been done that says that before this virus decays to where the patient will be free of virus, that will be about 70 years, right? So 70 years, obviously, something else will have killed you rather than HIV. So that is, we can't just use ART. We need something else if we need a cure. So about 12, 15 years ago, there were glimpses of hope of cure that fueled the cure, I'll call it cure research revolution. First, there was Timothy Brown. We can show his face here because he was a strong advocate for HIV cure. So he got cured when he had leukemia and had a transplant with CCR5 delta donor. So the virus needs this receptor to enter the cell. And so he got transplanted, and then he got transplanted again. And the way this transplant is done is that all of your T cells are ablated or removed, and then you get a new one from the donor. And over time, he subjected himself to all kinds of studies, and the HIV never came back. He died not too long ago from non-HIV related causes because leukemia, even when you get a transplant, can still come back. We also have the London patient, and then we got the Mississippi baby, who was a baby that was born. The mother did not attend antenatal, was found to have HIV. The baby was born. Baby was started on ART that was a bit non-conventional, higher doses than, more medications than usually done for babies. And then the mother was lost to follow up for many months, and then reappeared. When she reappeared, she had not given any medications to the baby, but the baby virus was still undetectable. And the baby was followed for many months, but eventually the virus came back. Now, what that tells you is that, probably if you're able to catch somebody when they get HIV and you treat them, you might be able to suppress the virus to the extent that if you stop ART, it will not come back. So this has been an important area of cure research. And there have been cohorts of people who were identified early and treated, and some of them are still free of virus. But for majority of people, the virus does come back because the reservoir gets established very quickly, usually within 72 hours. So how will an HIV cure look like? So in this field, we talk about eradication cure. We used to talk about sterilizing cure, but then we don't say that anymore because it's not very politically correct. And then we have functional cure. Functional cure is where, like, you may have some of the virus, some of the reservoir still present, but then even if you stop the ART, you will still be able to live without the virus becoming detectable. And then, of course, we have sort of a mixture of both. Let me run you quickly through HIV cure strategy so that when I talk about these things later, you will understand them. So there are a number of cure strategies that scientists are working on. The first and the most studied is what we call the shock and kill approach. So the idea for this is that you have the virus that is in these resting CD4 T cells, right? So, and the person is on ART, and these cells are not multiplying, or the virus is not multiplying, therefore you cannot get rid of it. The immune system also cannot see it because they are not making any envelope, they are not making anything to the surface of the cell for you to target, for the immune system to target it. So the idea for this is that what if you could use another agent? So another agent that we call latency reversing agent, you add it to the person with ART, and then the latency of the virus begins to be reversed. It begins to make viral proteins. And then the immune system can now get rid of this virus, or even virus cytopathic effect can lead to cell death. HIV, usually when it is multiplying, is toxic to the cells, right? Especially T cells. Not to macrophages, but especially to T cells. So then either the cell will die, and then eventually you can stop the ART. The idea also is that even if the cell makes new virions, because of ART, these new virions will not be able to infect bystander cells, right? So that is the idea behind the shock and kill approach. And there have been several studies done on this. There have been clinical studies done on this. There have been phase 2 clinical trials. What all of them have shown is that you will be able to get reactivation of the virus, but reducing the size of the reservoir is a different issue. So far, we've not been able to do that, but it could be that we are not able to reactivate enough of the virus in the places that it matters, or it could be that, the killing, we need a different kind of killing mechanism. So people are working on different methods to kill, the cells that are able to reactivate their viruses. So that's the shock and kill approach. And then the next approach is what we call the block and lock approach. So that is like the opposite of the shock and kill, in that for that, what you do is that now you have this virus that is, that is a provirus that is latent. So what if you can give something to make the virus super latent, right? Such that it is so much locked in the genome that even if you stop your medications, your ART, it will not be able to come back. In other words, if you can modify the epigenome around the virus and then enable the virus to be permanently latent. So it's not such a crazy idea. I'm sure some of you are looking at me and like, what are you talking about? It's not such a crazy idea because, the human genome is about 8% retroviruses. I don't know if you knew that, but there are retroviruses all over the genome that scientists have been able to actually get out of the genome and culture it, remake it and culture it in cells. So they are there, they are not able to replicate because of the epigenetics that is around it. Okay, so this approach has been moderately effective in mouse studies so far. And then the next one is gene therapy, right? So this one is straightforward, but not straightforward at all. Straightforward because it's easy to understand, right? What if you can use something like CRISPR-Cas9 or TALENs or any of those things? First, what if you can remove the receptors that are needed by the virus, like the CCR5 receptor, edit it, and then put it back into the patient? Or what if you can use CRISPR-Cas9, somehow let it target the latent reservoir, it will get in there, and then it will excise the HIV, and then you are free of HIV, right? So some trials have been done with CRISPR. Recently, it wasn't successful. The scientists were from Washington University, where I am. They think that maybe the dosing, they were afraid, right? So this is just the beginning, so you don't want to give tons of it to the patient. So they think that maybe the dosing was not good enough, but studies are being done on this. And then there's a bunch of monoclonal antibodies. These are new approvals per year of monoclonal antibodies that are in the field. And what people are doing now is, what if you add that to ART, will you be able to keep the virus in latency or keep the virus, get some kind of functional cure? These monkey studies show that when they gave this monoclonal antibody, over time, the virus was not able to come back after many, many, many weeks. So these are promising studies for functional cure. So as you can see, which of these will be appropriate for us in Africa? Like when it comes to shock and kill and block and lock, that is easy. You are just adding one other or a couple of other medications temporarily until you are able to stop ART. When it comes to gene manipulations, that is not that easy, because you have to take cells from the patient, manipulate them, put it back. So the logistics is quite a lot. And then stem cell transplant, that one, we won't even talk about it, because usually this is a very, very severe procedure. And if you're a physician and you go and see patients who have had stem cell transplant, a lot of them die from just the procedure. So for people who are doing so well on ART, you probably wouldn't do this unless they have need for it for another reason. And of course, the immune-based therapies, we think, can also be easily translatable. So that is kind of a whirlwind through the cure strategies. But if you look at the literature, right, you see that a lot of this cure research is being done in the US or in Europe. And Africa is left behind, right, in cure research. So that is what we sought to change. Somebody met me somewhere and said, are you the one who is trying to revolutionize cure research, to democratize cure research? And I said, yes, I'm the one. So why do African scientists and patients, why must we participate in cure research? First of all, most of HIV is in Africa, so I think that is just, that is just good enough reason. But there are questions about the HIV reservoir that can only be answered here in Africa, in my opinion. So there are co-infections with tuberculosis, with hepatitis, malaria, and all kinds of co-infections. All of these co-infections can alter the immune system. They can alter how the T cells behave. And therefore, if you don't take that into consideration and you get a cure, your cure might not be applicable to these patients. In addition, we have, HIV too in West Africa. West Africa is even more interesting than anywhere else in Africa, because in Southern and Eastern Africa, it's a little boring. You just have one subtype, C, or D, and then in the global north, it's mainly B. But in West Africa, we have circulating recombinant forms. We have ADG, CRF02AG, CRF02 CPX, all kinds of, this is just a short list. So we have a whole lot here in West Africa. So our epidemic is a lot more interesting. And so it needs to be studied, and it needs to be studied in cure research. Okay, go back. So in addition, I talk about all of these methods for cure, right? So now, when it comes to cure, these are people who are doing well on ART. It's not like somebody is going to die tomorrow. So if you are bringing them a cure, it should be something that is, in my opinion, relatively benign for them to be able to take the risk. So the question is, how will African patients accept or decline the type of risk that we want them to take when it comes to HIV cure? So that is an important question that needs to be answered. And some of these studies have been done elsewhere, and we have begun to do some of these studies. So this is a review that we were invited to write in Current Opinion about HIV cure acceptability. So, you can go to conferences and say things like, oh, very little of the cure research is being done in Africa, but where is the evidence, right? So you want to show that this is really true. We know intuitively that it's true, but we wanted to get some numbers for it. So what Evelyn in our group led us to do was to look at all the cure-related literature from 2010 to 2022. We excluded vaccine studies, other trials, or other, HIV studies, except it has to be cure research. Cure research has some specific characteristics that we know, so that's what we used. So we had, over the 12-year period, we had 1077 publications. As you can see, cure research has really ballooned over the period, right? So from 2010 to 2022, about 10-fold increase in cure-related research around the world. So that was interesting in itself. However, when it comes to where it is being performed, about most of it, so most of it is in the US and Europe, and a little bit in Asia. And Africa, we didn't come last, though. I think we were more than South America. but they don't have as much HIV as we do. And the number here, the percentage was 2.9%, right? So that is extremely low. We were expecting it to be low, but we didn't expect it to be this low. I guess that's the thing. We were expecting it to be low, but maybe 8% or something, but 2.9%, that is too low. And when we broke down the few studies that were done in Africa, most were done in South Africa, which is not surprising. And then West Africa, we had just a few. When you look at the funding, the NIH was funding most of it around the world, including the ones that were being done in Africa. They are also funded mostly by the NIH. I think other big funders in Africa, like Wellcome Trust and EDCTP, have not yet gotten on the HIV cure side of things. So, this goes to what Professor Andare was saying yesterday about we African governments stepping up to fund our own research. Because if you have 70% of HIV and somebody else is funding it, that is not so good. When we look at the various types of studies, so basic science is like what you do in the lab with cell lines and things like that, models and things like that. Again, very few in Africa. When we look at ex vivo, where you take blood from patients and then you manipulate the blood in the lab, we were 4.7%. Animal studies was 0. And then a lot more in the epidemiology side, and then also human clinical trials. There may be, one can come up with all kinds of theories about this. Is it because of equipment? Is it because of know-how, because of personnel? But I think a lot of African countries now have the tools that it takes to do this type of research. And so I'm encouraging the young ones here to get into it. So what is our solution to this problem? Our solution was the HIV cure research infrastructure studies, popularly called HCRIS, which we started here at the University of Ghana at the Noguchi Memorial Institute for Medical Research. We began by sending a couple of technicians to my lab at WACCBIP, and then since then we had a lot of exchanges. The aims were to, first of all, form a cohort of patients and characterize them for cure research. Now, the reason is that, like, if you have, like, when you are in Europe or US and you want patients that are fully virologically suppressed, because usually HIV cure research, you do it on those whose virus is less than 50 copies per mil or less than 20 copies per mil. Over there, you can just go to the clinic, look at the clinical records. They have regular viral load checks, so you can find out who is fully suppressed. But in Ghana, it's not like that. The viral load checking is very sporadic, and so you cannot really say that patient A is fully suppressed for the past 2 years or for the past 3 years. So we needed to build our own cohort and follow them over the years so that we know that when we are recruiting people for these type of studies, we know that they are fully suppressed. And then we also did some lab work and screened 150 epigenetic modifying compounds. The graduate student who did this is graduating tomorrow, so congratulations, Chris. And then we have also assembled a well-characterized biobank in our freezers and liquid nitrogen tanks. And then, of course, to use all of this to train people. So the result is that we've established all the laboratory techniques for HIV cure research here at the University of Ghana. So we can do everything from there to there, which is really good. We've trained several postdocs, fellows, graduate students. With the help of WACCBIP, a lot of our PhD and master students were trained here at WACCBIP, so we work together. So we are very grateful for the excellent training that they received. And then we follow this cohort to date, since 2019 to today. We follow them every 6 months. There are two of us managing the project, myself and Dr. Bonnie, or is she? So she's somewhere here. And then, yeah. And then we have about 6 postdocs or junior faculty. We've trained 5 PhD students, 3 graduated, 8 MPhils, 4 graduated. We have good number of technicians. Group now is about 22 people. And in terms of grants and fellowships, I was just calculating it yesterday. We've received close to $5 million to do this. I'm proud to say all of our postdocs have received one fellowship or the other, or gotten an NIH R21, or gotten something to do their research. So I'm really proud of what they've been able to achieve. And our PhD students also get various kinds of grants for their work. Going a little bit into what we've done, so if you look at the viral suppression among the cohort, it's actually pretty good. If you use the WHO, less than 1,000 copies as suppression, about 90% of them are fully suppressed. But if you use the international one of less than 50, initially we had a fewer, around 70%, but as time went on, it got better. So this is when dolutegravir was started here in Ghana, and that has made a huge difference in viral suppression. One of our graduate students who train here at WACCBIP look at viral reservoir size among those whose viral load was less than 1,000, between, let's say, 50 to 1,000, and those less than 50 copies. And the reservoir size is much bigger, about 3 times bigger, in those who are, whose viral load is between 51 to 999. That is the WHO accepted one. So I think the WHO should change the recommendation. I think they are doing that. Although the latest recommendation, they still hedged a little bit that it depends on the physician and all of that. But I think Ghana has decided to go with less than 50, so that is good. And we did present this data to the AIDS control program. So I'm happy they are changing it. And then we look at what Ghanaian patients think about HIV cure research, what risks they are willing to take. So these are all the things that usually you need to do with patients, and when it comes to HIV cure, you can see that when it comes to procedures, questionnaires and blood draws are easy for all the patients. But when it comes to, clinical trials and other things, not everyone wants to participate. When it comes to the amount of blood they are willing to give, you can see that the very few people don't want to give anything, but less than a quarter pint. When it comes to like a quarter pint, you have very few patients. Now, the reason we did this was that when it comes to HIV cure research, we don't take like 10 mils or 5 mils, right? So typically for our patients, we take like 100 mils of blood. Yes, so that is a lot of blood, right? So not everybody is willing to give that. But usually when they see that, we ask them, have you had a family member donate blood before? In Ghana, usually when you go to the hospital and you need blood, a family member goes to donate, okay? That will be news to some people, but that is what we do here. And so they know that. They know that, when you go and donate, they know how much you go to donate. So we tell them that is a pint of blood, right? And, so we have a container that we show them. This is how much usually people donate. This is how much we're asking you to donate. When we say that, they say that 100 mils is not a lot. So a lot of patients are willing to help us. But when it comes to treatment interruption, so if you are going to have a cure for HIV, right, you need to be able to convince patients to undergo what you call analytical treatment interruption, which means that if you're giving something that you think is going to cure them, then you have to convince them to stop their ART and monitor them closely to see if the intervention is working. So this has been done in Europe, US, and other places. In Europe and the US, when you ask patients about this, about 70% said they are willing to undergo ATI. Easy, right? But here, about 70% said, actually 67% said no way. Like they will never undergo ATI, right? And 17% said maybe. Okay, so that is a lot of people. What are the reasons for this? Is it because they don't understand? Is it because... So we did a more, you can see 2 studies there. We did a more qualitative study. And the reason is that the patients are telling us, look, I know people who stopped their ART and died, right? So, and so even if a doctor is going to follow me, I don't want to stop it. Okay, so in some way, this is really good, right? Because it means that the patients really trust the antiretroviral therapy because they know what it has done for them. But when it comes to cure research, we may have some trouble convincing them. So this is what some of them said. Said the boundary will be if they ask me to stop taking my medication, I'll need serious counseling on that. Okay, so and then what I was talking about is also over there. So in our lab, then here at Noguchi and at Washington University together, we've done quite a number of studies on the shock and kill that are published. We've screened a number of compounds. The latest one was what was done by Christopher, where he screened 150 compounds, epigenetic modifying compounds, where he identified 3 of them that can reactivate the virus from latency. So the idea here was to determine if we can get latency reversing agents that are more superior. I think the most superior one he got is this MC1568, which is better than vorinostat, which is like the standard in the field. These are 2 positive controls which cannot be used in clinic because they are extremely toxic. So the next step will be to do something like animal studies with these. And then, in relation to that, we've created a novel macrophage model for latency, because we realize that when it comes to T cells, it's very different from macrophages, where they don't respond to the latency reversing agents that are in the field. So we needed a model that will enable us to screen compounds for macrophages. So we engineered the HIV virus such that when we sort the cells on the flow cytometer and identify the red cells, those are latently infected cells that have not been reactivated. And then we can reactivate it with various compounds and then see how it goes. So we can do this. We have many clones of this cell line. Some of them are very easy to reactivate. Some of them, no matter what you do, you can't reactivate them. And this is very true in the HIV field, where even when you reactivate viruses from patients, there are some cells that will never undergo reactivation. Some we need to reactivate another time for them to undergo reactivation. So this model is going to help us to make waves in the macrophage field. We've also done work on block and lock, where a few years ago we identified this protein SF3B1 as critical for HIV replication. We found out that it interacts with the viral protein TAT here in immunoprecipitations, and we determined that this basic region of TAT was required for the interaction. As you can see here, the full-length TAT interacts in these E. coli experiments, but the truncated one that we truncate over here at 48 does not interact. So what we've done in the lab now is to create a system where we have portions of TAT, portions of SF3B1 that we've mapped for the interaction using this nanobit system, where if you co-transfect these, if the proteins interact in the cell, then it will generate luminescence for you. So with that system, we'll be able to screen for compounds for the block and lock approach. And I'm happy to say that it looks that the system is going to work. We did screen for a few protein kinase inhibitors. We identified this one here. And when we went into the cells to see whether it actually works, it's able to inhibit HIV reactivation from latently infected cells. So with this, if we get our next grant, we are going to screen for a number of compounds to see whether they can enforce HIV latency. So what are we doing next? So all of this are things that we've been doing here, and we wanted to enlarge this, right? So we've trained a number of postdocs, graduate students, and so on, but we want this to expand beyond what we have. And so we recently applied for, and we were fortunate to get an NIHD 43 training grant to continue with the training. So this is about $1.6 million that will enable us to train, to build capacity here at the university, and also with people from West Africa in HIV basic science and cure research. The program has program directors, myself, Evelyn, and Lian Shan at WashU. We have an executive committee. Peter is one of them. Prof. Kwe is another of the executive committee. And we have an advisory committee of the big people, Prof. Awandare and Prof. Yeboah Menu and a few others are on the executive committee. So we are very grateful for all the support because we think this is going to help. It's going to help us to train 4 PhDs, 7 MPhil, and 7 postdoctoral fellows in HIV cure related research. So these are some of the things that the fellows and students in HCRIS are doing. screening compounds for the shock and kill and block and lock approach, looking at drug resistance in people with persistent low-level viremia, that is, those patients that talk about with viral load between 50 to 1,000. Impact of co-infections like TB, hepatitis, malaria, Hermann's, HPV on the viral reservoir, because these are very prevalent conditions here in Africa, and we think that they are going to affect how the reservoir behaves. And actually, we already have data on tuberculosis, where people with TB co-infection, when you try to reactivate the virus, you have a harder time trying to reactivate the virus. And we think it's because their T cells are more, quote-unquote, exhausted than regular HIV patients without TB. We also have a good data on hepatitis that have been presented at the HIV Cure Keystone Symposium that was held recently in South Africa. And we have others working on malaria and Hermann's and HPV and how they affect the reservoir. We are also creating a new latency model for HIV too, because we think that if anybody is going to work on HIV too, it should be us, right? So it's a West African thing. So we are working on it. And then looking at how the various subtypes that I mentioned also may affect the reservoir, may affect latency. And then looking at how comorbidities and immune activation also may affect the reservoir. So our plans are to expand the HCRIS cohort to cover the whole country. Right now, the cohort is only in Accra and Cape Coast. So that is just southern Ghana. So we need to move to the north and other places. And we are also working with some colleagues in Senegal, Burkina Faso, and Nigeria to expand the cure research in those, to start cure research in those places as well. Because Nigeria, for instance, has a lot of different subtypes that we will be interested in. So the funders who are here, you know, we are looking for money to do all of this great stuff. So yeah, we want to position ourselves as a site for HIV cure trials in Africa. The thing is, if you don't characterize our virus, right, then if there are trials, and trials are coming, if there are trials, you can't participate because you don't even know how to check the reservoir size in your, because I didn't mention this, but for HIV cure research, you need to be able to measure the reservoir size. And the tools that are currently used is only for subtype B. So you can imagine there's a trial for, you know, for a cure. We can't do it in West Africa because the subtypes here, we cannot use the same tools, okay? So we are developing those tools, and I think we will be successful very soon. It's not easy, but we are almost there. And of course, we want to train more people. So in conclusion, I'll say HIV cure research is advancing at a rapid pace. And the International AIDS Society, NIH, everybody is on board. The approaches, the main ones are what I talk about: shock and kill, block and lock, gene therapy, and immune therapy. And almost all the cure research so far has focused on subtype B, neglecting the many subtypes in Africa, and especially in West Africa, where we have a lot of different subtypes. So we in Africa, African scientists, we must step up so that, you know, our patients can be included in cure research trials as they come. So to achieve this, we need to do a lot of training, mentorship, which we are happy to do. So I'd like to thank my team. We call ourselves the Massive Action Team, so we take a lot of action. And then all the hospitals that we've been working with. I want to thank WACCBIP for helping us train a lot of these students and all the funders that are listed. Thank you. Hope I didn't go beyond. Okay.
Thank you very much, Prof. Che. So I think we have time for a few questions. Leopold.
Thank you very much for a very exciting talk and a lot of kudos for all what you are doing. Two questions. First of all, you show that in your trial, you're able to suppress the virus to up to 50%, 50 copies. And I don't know if you have data on the performance on the different subtypes, because I don't think you show it. So that's the first question. The second question is, I realize most of your grant coming from the NIH. How are you affected by the new trend? And, have you considered, diversify your funded source? Thank you very much.
Thank you very much. So yes, for the less than 50 copies, it works for all subtypes. So the subtypes here in Ghana too, we have the O2AG, which is the majority subtype. We are still able to suppress it. So the ART, I think, works across the subtypes. There's some small, subtle differences for HIV too. When it comes to the funding, yes, we were afraid they were going to cut the D43, but luckily they didn't. So that is good. And we've had a bit of tribulation with some new grants that we are applying for. So in January, I sent in 2 R01s, which is like the big NIH grant, and then they were both withdrawn. I don't know. What that means is that they were not even reviewed, right? So because they were coming from, but I think they said they wanted some paperwork for us to put in the paperwork to say why we need to do the work here in Ghana and not in the US. So now that we put in that paperwork, they accepted to review it in May. So hopefully we'll get the money. So yes, so we have money from Expertise France, and we are looking for more money from them as well. And then we have applied. It's not for lack of application, believe me, but it looks to me that other agencies have not yet prioritized HIV cure. So maybe that is why we are having difficulty getting money from them. But we are not the type that give up, so we'll keep applying until somebody gives us money.
The Ghana Society of Herbal Practitioners, they normally have large, collection of compounds, which would help. I'm excited about your work because this is the time somebody is looking for internal solutions to internal problems.
Thank you so much. Yeah, so, people don't like to hear this, but HIV probably originated somewhere from West Africa or West Central Africa, and there are monkeys, right, that have SIV that are completely well. They have large amount of virus, but they are not sick. So some of us believe that, the cure may also be found here, and that is why we are interested in the herbal plant extracts as well. We work with Mampong Center for Plant Research. Yeah, so they plant medicine. So we like to work with them because they are scientific and they know what they are giving us. So we've been screening a number of compounds from them. Of course, I couldn't share everything here, but some of them are showing a lot of promise. So we'll see how it goes.
So thanks so much. For one of the first slides, you showed a viral decay, which I saw a log unit decline. What do you term as an appreciable decline? If I'm saying a log unit decline, or you need more?
Usually, so there are two things. There's the acute infection, right? And then for those who are fully suppressed. So for the acute infection, usually we get several logs reduction when you start your ART, and then that goes down to undetectable. For those who are on ART already, and when it comes to the reservoir, right? When it comes to the reservoir, a log unit in reservoir reduction is a lot. But when it comes to like acute infection, then usually we get several logs. It plummets, plummets several logs down when you start ART.
Okay, so I'm here. Thank you very much. Very beautiful work. I wasn't surprised when I saw that the reservoir size really is huge, even for suppressed patients. And I don't know how you're measuring your reservoir sizes and your latency model, but I never heard you mention clonal expansion as a major roadblock towards cure. And so, have you tried to look for these in these patients, and are you able to reactivate some of these clonal expanding cells? And which of these cells are really easy to identify in terms of their clonal expansions? Thank you.
Yeah, thank you very much. Those are very important questions. So clonal expansion is where you have the T cells, and then with one virus inserted, and then that T cell multiplies itself, because that is what they do when they need to attack, an incoming agent. So yes, so we are looking at it. We have PhD students now who are going to look at clonal expansion. Another reason why we should do this here is that clonal expansion in Europe or America, the mechanism may be different, right? So there are two methods for clonal expansion. There's homeostatic proliferation, and then there's antigenic stimulation. And antigenic stimulation is coming up to be the major one. And in the US, what has been discovered is it's mainly CMV. So a lot of the reservoir that are clonally expanded are CMV specific. Here we don't know what it is. Is it TB? Because about over 30% of our patients have tuberculosis. Is it even BCG that was given? What is it? So we are interested in figuring out what is responsible for antigenic stimulation here in our environment. And for that, initially we couldn't do that because you need to sort the cells with a cell sorter. But we recently got a cell sorter at Noguchi, so we are very happy, and we'll be able to begin some of those studies. Yes.
Yeah, it's great. Thank you, and congratulations on a wonderful program. Out of total ignorance, is there evidence at the moment to suggest that the latency reversing agents are affected by the subtype, whether there is enough evidence to know that that's true or not true at this stage?
Yes, so all of the clinical studies for LRAs have been done in Europe and the US. So we don't know whether, when you come to the patients, whether it will work for our subtypes. What we do know, though, is that we've taken samples from patients here, and we're able to reactivate the virus in the lab. So it will probably work here as well.
Just as you have been exploring the natural products, the medicinal plants, just wondering whether probiotics are also factoring in this, because they help build immunity and so on.
Yes, so probiotics and even the microbiome, right? So how does the microbiome and the chemicals that are released affect the reservoir? Even bile acids, right? How do they, and these are region specific, so the microbiome for people in our area may be different from people in Europe because of different exposures. So that is an area that we are interested in. We haven't put any student on it yet, but it's one of the areas that we are interested in exploring, to look at the metabolomics and things like that, to look at how that may affect the reservoir. Although we are doing some studies looking at how metabolites that are produced by macrophages may affect the reservoir, but we need to do a lot more on that.
So I have a question more along, we know that you can't reactivate every everything. So this is more of a, what do you think about, I think there's been some proposals, you do shock and kill to clear all those susceptible cells, and then you follow that up with lock and block. What do you think about that in terms of practicality and whether it's something that we could ever get to?
I mean, it can be done. For me, I believe that, even for the things that we think cannot be done, if you have a patient who is willing for us to try, I think we should try. At least we will know whether this thing can work or not, right? After all, it was this bone marrow transplant that enabled us to see that, okay, a cure is actually possible. And then people put money into it for us to do the work. So, so that idea is interesting. And of course, I didn't talk about a lot of things, but about chimeric antigen receptors and CAR T cells, and it's also a big one, trying to kill, the reservoir after reactivation. So there's a lot of that. And yeah, I agree that that can be tried as well.
Yes, thank you very much for the wonderful presentation. I'm just interested in your HIV cure using the super latency sort of treatment.
Using?
Super latency.
Okay, yeah. Mm-hmm.
Yeah, so I just needed a sneak peek of how you would think you would do this. Are you trying to tighten the accessibility of the chromatin? And then do you think it's also what really played a role in that immune baby that was first treated and they had some sort of latency for a long period of time?
Yeah, let me start from the bottom. So for the person who was cured, I think they were not even finding the virus. So, so that was, it looks like he had like a complete cure. If you are going to do a block and lock, it's going to be a functional cure in that you're still going to have the virus in there, only that it's not coming up, right? But you need to define what is around the virus, right? Is there something unique in the epigenetics around the virus that you can modify? So that is what people are actually working on now, trying to figure out, doing epigenetic, sequencing to see what is around the virus that can be modified. Because you don't want to modify everything of that sort if it is all over the genome, right? Because that is not going to be good for the patient in terms of side effects. So what is unique about the epigenetics around the HIV provirus? I think that is where the research is going now.
All right. Thank you. Let's give another round of applause. I think you all agree with me that this was a very, very rousing start to the day. Now we're going to start our sessions for the day. And I would like to call, before we do that, those who are queued to do turbo talks later in the day, if you haven't sent your slides to ICT, please do that before the coffee break or forever lose your place. Right. And then we will have a slight change later before the coffee break. We'll have some of our sponsors who were queued to present yesterday presenting. And then presenters, to avoid mishaps with the pointer, just point it towards the back. That's where the sensor is. So I'll call up the chairs for the first session of the day. Dr. Anastasia Akins and Dr. Winfred Seth Benwinyo to take us through.
Good morning and welcome to the first session of the day. Already we've had a good start with Prof. Che, and I'm sure we are going to continue in that same vein. So the first talk is a plenary session that's going to be delivered by Dr. Lily Pemka, who is a faculty member here at WACCBIP and at the Department of Biochemistry, Cell and Molecular Biology.
Thanks. Good morning. So my topic, here. Missing pieces, the uncharted genomic terrain of breast cancer in Ghanaian women. So with this talk, you're going to understand why I chose this title. Here's my... All right. So cancer is what you call a complex genetic trait. It's multifactorial. It involves both genetic and environmental factors. So typically what happens is during your life cycle, clearly you start off as one cell, the zygote, which undergoes so many divisions. You end up with a human being that's made up of billions of cells, right? And this process is tightly regulated, and they are controlled by genes, right? Now, when you have a healthy cell, the DNA is always replicated before it divides. However, when there are mutations in genes that regulate the cell cycle, this is compromised. So it gets to a point, there's overproliferation of cells. These cells do not respond to the normal cues, and then they grow bigger, grow beyond where they are found. Oh, sorry. Where's the light? Yes, it's working. And then it spreads beyond where it started. Now, when you have cancer in the breast, that's what you call breast cancer, right? And typically, when a cancer starts, it begins to spread beyond, and then these are the different stages. And so by the stage 4, it's going way beyond the breast into distant tissues, and ultimately, that's what kills you. So the breast cancer problem is one of the top cancers that are diagnosed around the world. This is a global issue. It's just not confined to Africa. You see the same case in Africa, right? This data shows at a point in 2020, breast cancer surpassed lung cancer as the #1 cancer diagnosed. However, it's dropped to #2 now. It's not different in Africa, and especially not in Ghana. What are the problems you are dealing with? The cases are increasing dramatically in Africa. When it comes to research, treatment, diagnosis, and post-treatment care, there's a vast disparity between Africans and then the high-income countries. Now, what I've observed is mortality is disproportionately higher. It's partly due to late treatment. However, if you look at the 5-year survival numbers, it's about 40 to 60 in Africans, and then you have 80 to about 90 in high-income countries. Now, when breast cancer is subtyped, based on the molecular expressions, you find that the triple-negative breast cancer, which is negative for all these receptors that are used for treatment, are absent. And then what happens is we have a higher prevalence of that. And with TNBC, it has a higher metastatic potential and is very difficult to treat. And it happens to be highest in African populations. What are some of the gaps in knowledge when it comes to breast cancer research? The data is really scanty. The epidemiology is not clear. Recently, I was just looking at the numbers of hormone receptor positive cases in our setting, and it was just a wide range from 20 to 80. It really wasn't clear. Even with TNBC, where we know is highly prevalent, the literature is just scattered, and it's not clear what the epidemiology is. When it comes to genomic studies, it's practically non-existent. Now, there have been some studies. However, they focus on certain genes, the most popular ones. I'm sure all of you have heard about BRCA1 and BRCA2. And so because of that, some of these are known variants that have not been studied in our population, and the environmental factors are not clear. When you look at genomic studies in general, worldwide, most genomic studies have been done in Caucasians, about 78%. Only about 2.4% have been done in Black populations. So we are significantly underrepresented. That means something. Okay. Now, why are we conducting genomics in breast cancer in Africa? Because there's a lot of research that has been conducted everywhere else, right? So why don't we just copy from that and not waste resources? These are some of the reasons. When you look at our genetic background, we happen to be the most genetically diverse population. So if you look at this image, for example, you're looking at haplotype heterozygosity across several populations. And when you move away from Africa, the diversity drops, right? Secondly, there's a study that was done in 2018. About 910 people were sequenced. And it turns out that once we were aligned with the reference genome, there were so many reference contigs that wouldn't match to the reference genome that we currently use. And they happened to find about 10 extra bases that were not incorporated into the reference genome that we use. And why are we the most genetically diverse population? Humans evolved in Africa over 5 millennia, until about over 100,000 years ago, when it had other smaller populations populate the rest of the world. So if you use data that's been generated primarily in European populations, the yardstick falls short of what you can use. Now, what are the other unique things that I can talk about here? When it comes to genetic disease, because of the difference in genetic background, it presents with racial and ethnic variations. Like I mentioned, the TNBC, which is highest in African populations. And we've experienced a unique gene environmental interaction. And if you're dealing with a complex genetic trait, the best place to actually determine the genetic underpinning will be in a population that has, lived and evolved in Africa. Now, when you look at some of the data that have been generated in other populations, you realize that there isn't a lot of overlap. There are a number of GWAS studies that, when were done in blacks, it was different. Now, if in this era of molecular medicine, where we are developing drugs based on genomics, what happens now is that because we are largely excluded from these studies, all these molecular therapies might not work very well with us, although we share 99.9% of our genome. But for example, when it comes to pharmacogenomics, which is really important when it comes to a breakdown or metabolism of drugs, we found out that Africans are more likely to experience adverse drug responses because most of the drugs have been tested in a different population, which is more selective for variants that are in that population. So now, because of the high genetic diversity, we have this unique opportunity to identify novel variants or genes that underpin some of the complex genetic traits. So now there's this need to have, conduct population-based study that can be helpful when it comes to diagnosis, treatment, prognosis, etc. All right. So the study rationale. I already mentioned some of the existing platforms that are being used for other populations are not necessarily relevant, especially the targeted ones. So with whole exome sequencing, you're providing a larger platform to identify some of these unknown variants. And so the whole goal was to identify germline and somatic variants that are underpinning breast cancer in our population. Today, I'm going to just focus on just the germline part and talk about somatic stuff when we run into each other. So this is what we did. All right. So, of course, we acquired ethical approval from Gucci, EGBAS, the Kolebu Teaching Hospital, the Ghana Health Service, and we recruited 114 patients. Some were on treatment, some were neoadjuvant therapy. Some were treatment naive, and some were on neoadjuvant therapy. So we collected saliva, tumor adjacent, normal. We didn't take tumors from those who were on neoadjuvant therapy because of the changes that the drug would induce in the tumor, right? And then we conducted whole exome sequencing followed by data analysis. So this summarizes,
Let's use the pointer here. Point to the back.
Where? Okay. The back.
Yeah.
All right. What's happening? All right. Out of the 144 cases, 14 of them were saliva, 77 tumors, and 23 non-neoplastic tissues. Clearly, we can't get normal tissue, so we resort to getting tissue that's adjacent to the tumor, which is considered non-neoplastic or in quotes normal. And then for controls, owing to the lack of genomic studies in our population, it's very hard to come by, controls, especially population, relevant controls. So I think Elvis talked about the hearing impairment study yesterday. We used some of their control exomic data for analysis. Now we collected data from all over the country, and if you look at this summarizes the ages from 35 to over 60, and these are the histological subtypes. And then look at the stages. The highest was stage 2, and then when we look at the grades, the highest numbers were at grade 2. So we collected data on their, ethnicities, hoping to see if there are any, ethnic variations. However, the numbers were not as big, so we didn't do any meaningful analysis. Yesterday, I was at another meeting where a lady had mentioned that TNBC happened to be more common in the GANs, and she didn't know why. And this is the thing, the variability is so high, and then when you have different populations, it's likely some of these variants have become common. And therefore, if they underpin conditions like this, you are likely to see some variants that are more common in certain groups. So now, we found, following the analysis, we found 20 pathogenic germline variants in the cases, and we saw 5 in the controls. And then we had GJB2 overlapping, but the 19 were unique to the breast cancer cases, and they had 4 that were unique to the controls. Now, if you look at the breakdown in CLINVA, from likely benign to variant of unknown significance between the controls and then the cases and the controls. And then we looked at the variant consequences in the controls versus the cases, from frame shift deletions, frame shift insertions, unknown, non-synonymous, and then stop gain variants. So out of the 20, we found 7 novel variants that were associated with breast cancer in the cohort. From a new BRCA mutation, in this situation, it was a 5-base frame shift deletion. We saw new exonic variants in PALP2, FUS, HBB, and so on and so forth. So we had about 7 novel variants that we found even in this small population. Now, so the key findings. So using whole exome sequencing, we found novel population-specific variants beyond the known BRCA variants that are typically seen. And out of the 20 pathogenic one, I mentioned the BRCA2 frame shift, it hasn't been seen before. Now we have new 9 candidate genes. They've been associated with certain cancers, but not necessarily breast cancer. And then the 7 variants I mentioned. And looking at the functions of the genes, some are linked to drug resistance, tumor progression, immune modulation. And this basically underscores the need for functional validation, because in a Ghanaian-specific model, because these are, of course, when you find these novel variants, you need to conduct some of these functional studies to figure out if it's actually associated with the condition. And so we highlight the value of broad genomic approaches instead of focusing on this targeted sequencing, and then including Africans in cancer research. Now, I mentioned how the mortality rate was disproportionately higher. I mean, there's so many reasons behind that. Sociocultural beliefs, low awareness, and of course, some of these subtypes that are more aggressive. So conducting research on its own, I mean, it takes a while before it becomes, quite relevant or impactful. So in the absence of that, we observed that low awareness is quite low in the Ghanaian population. That was probably contributing to the late presentation. We ultimately contribute to the higher mortality rate in our population. So we established the NUFO Festival. These are breasts. I'm sure everybody can see their breasts. And basically, it's been running for like 7 years. And what we do is to conduct free examinations for these women, teach them how to conduct their own self-examinations. We've had thousands and thousands of women screened. We do on campus, we go to marketplaces, and these are a few pictures. We try to make it a bit fancy. So this is the very first one we had these cakes made. And then the other ones on the right, we believe in diversity. So you can see all the different shades of breasts, colors. And then these are more pictures. If you are looking for Professor Wandari, he's here. is happy to support something like this. We've been working throughout COVID, and of course we'll have it this year as well. All right, so first of all, I'd like to acknowledge Dr. Claudia Anyigba, my first PhD student. She graduated last year. She's now a lecturer in one of the universities in the north, and she did most of the work on this. Dr. Lucas and Amega Etego, yes, one of the supervisors, and then all these people, including my lab, the participants who magnanimously donated their samples, collaborators at the Kolubu Teaching Hospitals, and WACCBIP for the funding. I got the Delta's funding, and that's what helped me relocate and start my lab here at the department. Thank you.
Thank you, Dr. Pemka, for keeping to time.
I did it. I was trying to.
Yes, so please, questions. Okay, there's one at the back.
Thank you very much, Lily. That's really interesting. Have you tried to look at the HER2 landscape to see to what extent the Ghanaian population may be carriers of HER2 and how that may be impacting therapy?
So the HER2 landscape, I didn't hear the question. Have I looked at the HER2 landscape?
Yes, I'm asking whether you looked at the HER2 landscape in the Ghanaian population as part of your study, since that's been established as a major pathway to treatment failure, and whether that has contributed to clinical failures in many of the women that we have, because you mentioned that disproportionately high mortality compared to other parts of the world.
Yeah. Okay, so yeah, good question. We've not really followed people with HER2, but even if you look at our samples, right, we do the sub-classification, the molecular subtypes. There's a number of them who are HER2 positive. If you look at the somatic variants we've identified, I didn't show that here. We've seen some variants, but we've not really followed, patients on HER2 to figure out how it impacts their treatment. We've not really done that yet. Does that answer your question? Yeah, we've not done that. We've looked at the actual HER2 mutations. No, we had to look at that. We've seen a couple of them in the samples, but we've not really looked at anything novel. Yeah, but it's a good question, especially if they're using antibodies at this point, and if those antibodies are going to be compatible, with the variant in our population. Yeah.
So. Yes, so the 7 novel genes that you came up with, very interesting. Are they related to Ghanaian population, or, are they new to Ghanaian, or is new to the world? That's one.
So some of them are novel. Okay, all right.
If you want to characterize them, how are they related to pathogenicity and so on? Where do they stand? How novel are they?
Yes. A few of them haven't been seen before. So at this point, we're relying on scores like CAD, SIFT, polyphen, to estimate the deleteriousness of those. So for those ones, until we do functional work, we really don't know the actual impact. The couple of them that have been seen before, there's one that is associated with ALS, but it's been found to be expressed in breast tissue as well, but hasn't been really explored. So until we actually go in and do a functional characterization, at this point it's an association. And we are largely relying on, like I said, in silico tools like SIFT and polyphen and CAD scores. So until we do that, we wouldn't really know. But at this point, they are really good candidates, Yeah.
Okay, so Lily. Yes, so I think you've done a comprehensive brain study on your work, on your research, but my interest is on the breast cancer. In terms of your awareness creation, have you physically encountered someone who has breast cancer in terms of how it looks like, and after treatment, how it also looks like? So, and also in terms of the churches around here, we have churches, Anglican Church, we have LIC, we have Catholic Church. Have you been there to in terms of expanding your market in knowledge about breast cancer to the churches and also to the ministries, yes, in the offices. I think you should. You are a scientist, yes. You are a scientist. You should, probably have your market there too.
Okay.
Yes.
All right. So let me answer those questions. So I've seen people I've seen scars and all that. I've seen before and after pictures, but I don't. Yes, I've seen that. The one good thing about the awareness program is a little different from what you see. A lot of people make noise around October, but we have a really richer, how do I say, expertise. We have the scientists, we have the nurses, we have survivors so even one of the pictures, you see a group of survivors, or sometimes they even show you their scars. We have psychology, we have genetic counselors. So the whole spectrum of people who can help, come together and then raise awareness. Now, going to churches, yes, we do go to churches. I haven't been to this church, but we've been to other churches. And every year, we do conduct some of these outreaches at churches and provide screening for them. Sometimes they even approach us. Because in October, there's a lot going on. And we can't, of course, we can't cover it. There are so many people doing things, and they do approach us, and we do go there to render our services. So, yeah.
Okay, so the last question. Okay, Dr. Sapon.
Oh, sorry. Dr. Pemka, with regards to functional studies, which I don't know, is there a timeline to do those functional studies? And back to Dr. Harrison's questions. For my lab, when I was doing my PhD, there was a mutation that we found, which was a valent to leucine. And you think about it, it's a hydrophobic change, meaning that it's something that you might probably skip because it's valent to leucine. It's not like valent to aspartic acid, right? So a lot of us ignored it, but then we did a functional studies, and the phenotype that it produced was completely crazy. When you take HER2, it's about 20 to 25% amplified in a lot of these breast cancer situations. So I don't know if you have a timeline for these
functional studies
so that you can really nail down on what these mutations and the impacts that they're having on.
Okay, the timeline will depend on the money you have.
Okay.
So, thank you. Peter answered the question. But I have a master's student who's going to start, it depends on the funds you have, start slowly. So I have a master who actually prepared a proposal and all of that. And she was going to focus on some of the new BRCA variants, what we've seen and what others have seen. Now, what's happening around the world is people are getting excited about African genome. So they conduct the whole exome, whole genome, they do the association, and they leave it at that. At that point, it's not very useful because you don't know the impact, you don't even know how you can use that during genetic counseling because it's not determined. So my student is going to start off with, BRCA mutations, for example, and then do, all this in vitro, starting with cell lines, of course, what we have, and CRISPR, and then figure out changes in homologous recombination, knowing that BRCA is involved in that pathway. And so there's no time. Once there's money, the time will be very, very, very short. Okay. But yes, but we'll start off, if anything, we'll start off with cell lines. You need to prioritize some of these variants. You don't just go all the way to maybe an animal model, but you prioritize them, cell line, go further. If you look smaller animal models that you can use, and then go further.
Yeah. Thank you very much, Dr. Pemka, for the very insightful and exciting lecture. We will continue the discussion during coffee break time.
Neutralization antibody in clinically confirmed cases from the ongoing measles outbreak in Liberia. I am Kalilu, a PhD fellow at WACCBIP. Measles is a highly contagious airborne disease caused by measles virus, which is an RNA virus, and can result in serious complications or fatality. And it is considered as one of the most infectious hemopathogens on Earth, with a basic reproduction number of 12 to 18. And globally, in 2023, we have about 107,500 lives were lost. And those was mostly children under 5 years. And the virus is transmitted from person to person via respiratory droplets produced when infected people cough or sneeze. And the incubation period is typically around 7 to 21 days. The symptoms include high fever, cough, runny nose, and rashes. And measles-containing vaccine was first introduced in the 1960s, followed by the 2-dose regimen in 2009. And the first dose is administered around 9 to 12 months, and the second dose is also is administered as well, 15 to 18 months. And this vaccine usually gives around 97% protection. And also, the maternal-derived antibody can protect infants about 6 to 9 months on average. And the global distribution of measles virus, globally, WHO has recommended that you assign genotype based on the H and the N gene coding sequence. And we have about 8 clade with 24 genotypes. As you can see, the global map there, you see Africa, you have mostly is B3 and D4 and the D8. And B3 is the most frequent detector in Africa and consists of 2 clusters, 1 and 2. And D4, D2, and D8 is mostly frequently detected genotypes in Southern and Eastern Africa. Coming to the clinical course of measles virus, basically, measles virus has about 4 phases from the exposure to resolution or recovery. And the first phase, the incubation period, will last from between 7 to 21 days, and followed by the prodrome phases that is characterized by high fever, cough, runny nose, and reddish eye, and which is also, it lasts for around 2 to 3 days. And that's followed by the spotting, and this spotting is a tiny white spot that is found inside the mouth and lasts for 3 to 5 days, and then follow the rash and then the recovery phase. And measles in Liberia, as of December 31st, 2024, we have about 13,293 confirmed, and of those confirmed cases, 95 deaths were reported across the entire country. And of those numbers, 5.26% were laboratory confirmed, and 69.26% were clinically confirmed, and the rest was epi-linked. And measles-containing vaccine was first introduced in Liberia in 1978, followed by the second dose regimen, which is the MCV2 in 2019. And the MCV1 coverage in 2016 was 80%, and in 2016, and 96% in 2022. And the MCV2 coverage was 50% by December 2019. The rationale of the study, we said there's despite modest gain in measles vaccination coverage in 2024, more than 30 million children remain unvaccinated, and outbreaks are on the rise. And there was report that 60 countries experienced large and disruptive measles outbreak, nearly doubling from 33 in 2022. And serious survey provide a broad snapshot of past infection and successful vaccination, while neutralization assays assess functional capacity of the IgG to prevent infection. And it also provide a comprehensive understanding of the population immunity, which is critical during an outbreak. And Liberia remains an endemic for measles and experiencing measles reemergence due to suboptimal vaccine coverage rate, leading to rising clinical confirmed cases. Therefore, understanding the IgG profile in patients is critical to public health response and assessing vaccine effectiveness. And the objectives of the studies is to determine the level of measles-specific IgG in patients, and also to determine the neutralization burden of those IgG against B3, D4, and D8 genotypes. Methodology, the study was a cross-sectional study, and ethical approval were obtained from the Universal Liberia IRB and the Noguchi IRB, and also informed consent and assent form and questionnaire were administered and obtained from participants, and samples were retrospective and prospective samples. So here is the demographic characteristic of my participant, and as you can see in the figure, majority of my participant was female, which is 118 or 199, and 100 patients were vaccinated, 17 were unvaccinated, and 82 vaccination status were unknown. And 38 patients were IgM positive, while 161 patients were IgM negative. And coming to the first objective, And this objective were achieved using the commercially available ELISA kit. This kit is based on indirect ELISA. So after carrying on the first objective experiment, and this was the objective, this was the result that we obtained. And the result, we thought to look at the age and vaccination status, because these are the key two components when we're doing this kind of study. So when comparing the vaccination status between looking at the IgG levels, there was no statistical significant difference between the vaccinated and unvaccinated as it relates to IgG levels. And also, there were also no statistical significant differences among various age grouping compared. And putting this data together, and this will indicate that vaccination does not correlate with higher IgG titer for this study. And the level may remain consistent across age group, possibly due to similar exposure to measles and or vaccine outcome. Then we went on to look at the seroprevalence of the entire study, and we got 69.85% as a seroprevalence rate for the study, and the seropositivity was higher in age 11 to 20 years, which was 27.3%, followed by 5 to 10 years, 23%, and the lower recorded in those aged from 31 to 56 years. And this result indicate that immunity was higher in 5 to 20 years old, but dropped significantly in 31 to 56 year old due to possibly one immunity or missed vaccination, leaving 30% of the population susceptible. Because as you may be aware that because due to measles viral transmissibility, the WHO has set a cap of 95% as a herd immunity threshold, and this study was below that. And the second objective was to look at the functional capacity of those IgG, and this experiment was done using the pseudovirus-based assay. And so what was done was we were able to produce a pseudoviruses for all the 4 genotypes: the Emoston, which is the vaccine screen, the B3, the D4, and the D8. Those genotypes were produced, and then we'll do the experiment. So the result here show the neutralizing antibody titer exceeding protection threshold. As you can see, the first figure on the left there, we try to compare the neutralization titer between genotypes, and we found that majority of our participants were able to cross-neutralize all the 4 genotypes, but there were differences when comparing the vaccine screen and the B3, and also there was also the D8 genotype higher neutralizing titer than D4, and the data indicate that while most individuals were protected, they had protected antibody titer, but it was genotype-specific immunity variation was existing in this data. And the vaccine neutralizing antibody responses may not equally be effective against all circulating strains as per our data. And in the left figure there, the dot represent the protected threshold. All those individuals that are below this one, they were not protected against the genotype. Those that are above were protected against the genotype. And then we went forward to look at each genotype and look at each participant, group them into vaccination and non-vaccinated. And when we did that, we found that for all the 4 genotypes in the study, there were no statistically significant differences in neutralizing antibody titer between vaccinated and unvaccinated patients. However, a good number of unvaccinated individuals were above the protected threshold, which might be due to natural exposure. And this may imply that vaccination did not result in significant increase or decrease in the neutralizing titer when compared with unvaccinated across the 4 genotypes.
You have 1 minute more. 1 minute.
Yeah, we went further to do the correlation between IgG levels and neutralizing titer. And we found statistically significant difference between neutralizing titer and IgG level, and the association reached from moderate to strong correlation. And these findings indicate a significant and measurable correlation between IgG level and neutralizing titer in study participants. And then we went further to look at the correlation between age and IgG levels and the neutralizing titer. And we found no statistically significant difference between the titer, the age, and the IgG level. And the association reached from weak positive correlation to no correlation. And to summarize, the IgG level remained consistent across age group indicating similar exposure and vaccination outcome, and seroprevalence was 69%, and the higher was recorded in 5 to 20 years, and there were no statistically significant difference in terms of correlation. And we have substantial number of unvaccinated individuals have antibody above protected threshold, likely due to exposure. And the strong to moderate positive correlation was there between IgG level and neutralizing antibody titer, indicating these measurements were significantly linked.
Please be rounding up. Your time is up.
I'd like to acknowledge my supervisor, supervisory team led by Professor Kusi, and the course supervisory team was Tarakwachi. And also, I want to thank WACCBIP for the funding and these various center for providing me the space to do my work.
Thank you.
Thank you.
Yes, thank you very much. Questions, please. Questions? Okay, so as people get ready to ask questions, maybe I'll ask this one. Perhaps I missed it, but for all these subjects, the ones that you gave the statistics in the earlier slides, are these all children under 5, or what's the age bracket that we are looking at?
The age bracket is from 1 year up to 56 years.
1 year up to 56?
Yeah,
Oh,
they were.
okay.
Yeah, it was mixed. Yeah.
Okay. Thank you. Questions? Okay. It looks like we all well understood what you presented. Thank you very much. Okay. So the next person is Christopher Abana, one of Prof. Che's students who is graduating tomorrow. And he's giving us a talk on... Okay, Christopher, you're welcome. You have 12 minutes.
Thank you. All right. Good morning, and I'm happy to be back again. The title of my presentation is Acquiring Latent TB. Where's the pointer? So the title of my presentation is Acquiring Latent TB Leads to T-cell Exhaustion and a Reduced Reactivation Potential in People Living with HIV. So already my boss have already said much about HIV, so I'll just go straight to, we know that the most important cells that are infected by HIV are the CD4 T cells. And usually when these CD4 T cells are infected by HIV, a lot of them will end up dying due to the virus replication in the cell. Some will die, and there will be a few number that will remain to maintain the immune system that we talk about being homeostatic proliferation, or that will be maintained by this small number of cells. And this small number of cells will kind of grow. This small number of cells will kind of grow, and this is one of the major obstacles to finding a cure to HIV, in that even though these cells are silent, they are not replicating, at the moment, they are replication competent. There will be times that they will just wake up one day due to the antigenic stimulation and start producing HIV. And this has been one of the major barriers to finding a cure to HIV. And when we talk about HIV-TB co-infection, close to 70% of African population, as you can see there, are co-infected by both HIV and TB as well. I must say that TB is one of the most important opportunity infection for HIV. You can see majority of African continents infected by or affected by HIV TB co-infection. Only a few countries have minimal HIV TB co-infection. So we know that as during the infection of HIV, CD4 T cells are the most affected cells, and some of them will die. And we have a few number of them that will be remaining during the infection of HIV. And then when TB sets in, it makes even the case even more worse because we have very small number of CD4 cells that have been left to combat the immune system. So the immune system becomes so weak that nothing can go on, and it makes the infection of HIV progresses faster, and we have a lot of people dying from HIV TB as well. And the TB infection continually produces antigens that like kind of significantly contribute to the persistent immune activation in HIV. These antigens could stimulate T cells that result in more viral seeding, because even HIV alone will result in a viral reservoir. So when TB also sets in, it increases the viral reservoir of the patient, and it makes it very difficult. Like there's double trouble. Oh my.
Double trouble.
Yeah, so there's double trouble. So the person has HIV and TB at the same time. So the reservoir is a bit large. And the problem is that although the effect of HIV on the TB cells have been well characterized, we can see the same for how TB infection has effect on CD4 T cells in the context of immune activation and even exhaustion markers. So furthermore, also there's no the effect of TB or the effect of co-infection on the reservoir is not well characterized, or it's not well explored. So our aim was to determine the effect of TB on TB co-infection on activation of state of T cells in co-infected patients, and then the response of T cells to latent vaccine agents, and then we look at the reservoir of the virus. The research question is that, does persistent exposure to TB antigens affect the reactivation capacity of the resting CD4 T cells? Do we hypothesize that proviruses in CD4 T cells from co-infected patients will be more difficult to reactivate because they have a larger reservoir size, because they have both HIV and TB co-infection at the same time? So we have so recruited from our ongoing, already established cohorts that we have in our lab, that's run from 2019 to 2024, and most of these patients are virologically suppressed on ART for more than a year, less than 50 copies of viral load. And out of them, we were able to determine 37% of the patients having both HIV and TB co-infected individuals in our cohort. And then out of these people, we took 13 of these paired samples, 13 HIV TB cases, 2 healthy donors, 7 as negative controls for the rest of the studies. They also used 2 paired HIV TB co-infected patients for the to check for the reactivation of HIV in both either HIV only or HIV TB individuals. Yeah, so we isolated PBMCs. We looked for the also isolated CD4s, CD4 T cells, and CD8 T cells. And then we plated these cells at 500,000 cells, and we stimulated them with PMA ionomycin. We incubated them at 37 degrees Celsius, and then we stimulated the cells, and we did flow cytometry to look for the activation markers and exhaustion markers, as well as we went ahead to isolate HIV RNA from the CD4 T cells for the reactivation studies. So this is an ongoing study, so I'll just show you a bit of the data. My other colleague will show the rest of the data tomorrow, hopefully. So we will see that both expression and activation markers, both activation and exhaustion markers of the CD4 T cells and CD8 T cells are similar. As you can see, CD38, CD69, those are activation markers for the CD4 T cells, and PD1 and TIGIT are all similarly expressed in both pre-stimulation, before they were stimulated with PMA ionomycin. And then for the CD8 T cells, we can see similar expression of both the CD8, CD38, and CD69 activation markers, and the PD1 and TIGIT exhaustion markers as well. So after post-stimulation, after they were stimulated by PMA and neomycin, both the CD4 T cells, we could see similar expression of both activation and exhaustion markers in both type of cells, both CD4 and CD8 cells. However, we could see elevated levels of the CD69, and then in the CD4 T cells, as well as that of CD8 T cells, showing a similar expression of both activation and exhaustion markers. So we went ahead to look at the impact of HIV-TB on the ability to reactivate HIV from latency. Yeah, so we could see that. So these are two different patients paired with HIV, both HIV and HIV-TB. You could see from the board that we have patient 3018 and that of 2006. You can see two different patients, the HIV only and HIV-TB group. HIV only group is the one colored in blue, HIV-TB group is the one colored in red. We wanted to see the effect of reactivating HIV from latent HIV from this group of patients. So we stimulated them with our already known LRAs that we have in our lab to see whether it's difficult or easier to reactivate HIV from latency from these two different groups of people. So we could see that from both HIV, the first experiment and second experiment on to my left and right, could see that the blue bar represented those that have HIV only, because the reactivation of those people are easy as compared to the red bar that is the HIV-TB only. So it explains that it's difficult to reactivate those who have HIV from those who have HIV-TB only group than HIV only. So there was a limitation from our study. We were unable to determine the duration of latent TB among the participants, and this could have kind of clarified the impact of TB chronicity on T-cell immune dysfunction in the HIV persons. T-cell stimulation was conducted using non-TB specific antigens. That's limiting the effect of TB antigens in HIV mono-infected versus HIV-TB infected individuals. Also, the sample size for the reactivation studies, we use only 4 samples. We intend to screen more patients on that to get a clearer picture. We only use a smaller size, that is 4 samples alone for the reactivation studies. So our data so far suggests that there's concurrent expression of both activation and expression markers in HIV-TB co-infected individuals, suggesting immune dysfunction, and this does kind of potentially impact the reactivation potential of HIV-TB co-infected individuals. And this presupposes that for the HIV cure drive that Prof. Chi previously spoke about, if we want to include those co-infected with either HIV-TB or even malaria, we will have to have more aggressive latency reversing agents or alternative cure methods for this group of people. So these are some selected references that I use just to scratch the surface of our work. My colleague will tell you more tomorrow. And I want to say thank you to everybody, our lab team, and our collaborating university, and our patients will take our samples, and then the hospitals as well. Thank you so much.
Thank you. Thank you, Christopher. Questions, please? Okay, so Prof. Nils Kwashi, Dr. Kwashi, and then, okay, so you first.
Thank you very much for an excellent presentation. In your limitation, you really addressed a lot of my question, but I would like to first know, how did you diagnose the latent TB? Because, there are different ways to do so. And, you use PMA and neomycin to do the stimulation. These are very harsh stimuli, and I don't really agree with your first conclusion, because you did not really find differences in the markers between TB, HIV, and HIV alone. So it really makes it very difficult to really understand how, you conclude that, the apparent lack of reactivation that you find in 4 patients is due to exhaustion in T cells in TB, HIV. So first of all, how do you diagnose their latent TB? And secondly, unless you are using TB-specific antigens to stimulate those cells, it's very difficult from using, PMA inomycin, which is a very harsh stimuli, to draw such conclusion. Thank you.
Okay, yeah. So we use the interferon gamma release assay, the kit, to determine the latent TB infection of those people. Yeah, so like I stated earlier on, we didn't use TB-specific antigens, but we used the PMA inomycin. And PMA inomycin is a known or very robust activation agent that can be used for studies like that. So that's why we use those kind of PMA inomycin for that activation. Even though we know that we could have used going on to use TB-specific antigens in our next study that I would like to do. Yeah.
Thank you. Prof, question? Oh.
Okay, then do I speak without the mic?
Yeah.
Yeah, so honestly, I was alarmed when I heard that there is a super latency in HIV. And also there is a super resistance in malaria. So when Omega means Omega, it's possible here in our part of the world. What is likely to happen? I'm not asking you, since when God is here, you don't ask Archangel Michael to answer a question. So I will ask God himself to respond to what the dynamics may be if Omega meets Omega.
Okay, so God is here, God will respond. Okay, God.
So you want Prof. Che to answer? Okay.
Yes. The microphone, please.
All right, so we haven't done those studies for malaria. We haven't done those studies for malaria, so I cannot say anything about Omega versus Omega. But one thing I can say is that everybody gets malaria in our environment, right? So I've been asking some of our immunologists, how do you determine, is there a marker to determine somebody's past malaria burden? If we had such a marker, then that would be easier for us to say that, okay, you have had more malaria than me, and or this patient has had more malaria than this patient, and so how does that affect the T cells and their reactivation potential? And to the last question, I think, yes, the right way to do this, or the best way to do this, is to use TB-specific antigens, because PMI inomycin, you've been discussing in the lab, is too strong, and you are going to get, it's probably why we are not seeing any differences. So now that we know we are not seeing any differences, we have to come down a notch with TB-specific antigens, and I think that will probably show if there are any differences at all to be seen.
But are they available, the TB-specific antigens?
Yes, we can get those. We can get those from some sources.
Yes, Dr. Kwashi Peter.
Yes, so I think my question was sort of along the same lines, because I was wondering if I missed that I didn't really see a difference in exhaustion between the TB and the singly infected, which was a bit surprising. So I think it's along the same line of this question.
Thank you.
Yeah, maybe I also have a question. I wanted to find out the HIV, is it any type of HIV? I mean, are the exhaustions, are they the same, the levels for both types of HIV, the two main types?
So this is for HIV type 1.
Okay.
So we're here to do HIV type 2, HIV type 1 and 2. Okay. Yeah.
Thank you.
All right. Thank you so much.
Just wondering whether the people you were looking at, were they still on ART?
Yes, they were.
Okay, thank you very much.
Sorry.
Thank you. We'll continue during the coffee break. Thank you, Christopher, and congratulations for tomorrow.
Sorry, and before the coffee break, we are going to have some of our sponsors. We'll have Dr. Kwashi. You come. Oh, okay.
Yeah.
You take care of that.
So before the coffee break, we shall take 2 quick messages from 2 of our sponsors for this year's research conference. So I would like to invite Nationwide Mutual Health Insurance to the podium to give us your presentation. Thank you.
Good morning. My name is Samson Abuaje from National Medical Insurance. Okay, so someone will ask, who is Nationwide? So Nationwide Medical Insurance is a private health insurance. So what we do is that we provide healthcare for individuals and their corporate institutions. So we've been in existence since 2003. As I said, it's a private health insurance company. So we have over 85,000 subscribers and partner with over 700 HSPs nationwide. So HSPs basically means healthcare service providers. So they help with the administration of the healthcare needs. In the industry, we have 22 years experience. So with expertise and then the knowledge, Nationwide has a lot when it comes to helping corporate institutions and then individuals to deal with their healthcare needs. And then one of our ambition or the foundation on which Nationwide was built is customer oriented. So what we do is that we listen to our clients and basically try to help with their needs. So with every corporate institutions, what we know is that companies are guided by their core values and then their principles. So basically this is what direct our service or our line of operation. So integrity, technology, innovation, we are dependable and then efficient as well. So because of our expertise or our experience within the private insurance environment, we've clocked certain awards that we can see. So we have a lot of awards, but because of the presentation, we can't present everything here. So basically, these are some of the awards that we've chalked because of our experience. Okay, so with Nationwide, we have 4 services that we provide. We have the corporate, the retail, the third-party administration, and then the international health insurance. So companies can go in for the corporate plan. And then, you know, when it comes to the company, their rules and regulations, most of them will not put on all the dependents for the employees. So basically, some dependents like the parents, like the extended family members, will not be placed on the corporate policy. So those ones will go on the retail package. Okay, we also have international coverage that allows clients to go outside the country to also have their medications catered for. And then we have the third party. So the third party is a bit different from the norm or the traditional insurance. So that one, I wouldn't go much into the third-party administration. Okay, so we have certain clients that we are currently dealing with or we are providing healthcare for. You can see that we have companies within the various sectors. So we have the government agencies, you can see PA, you can see public service workers union. We also have the educational corporate clients like the HSC, Ghana International School. We have Lincoln over there. So these are a couple of our clientele that we deal with. You can see GRA is also there. So hopefully, in our next presentation, we are hoping maybe we have certain corporate institutions in here over there. So these are what goes into the product. Okay, so we have the medical consultation. So either the general practitioner or the specialist consultation. Investigation is also covered. Investigation has to do with the labs and then the x-rays. Okay. We also administer prescribed medication. So for health insurance, we only deal with prescription or prescribed medication. We don't do over-the-counter purchase. Dental is also covered. Optical is covered. When it comes to our admission, it's also covered within the policy. We do surgery, and then our females too have the opportunity to do delivery through maternity. Chronic care is for those on chronic management. So if you have any chronic condition, we also have room for those our clients or those clients who have chronic conditions. Emergency is also covered, physiotherapy, and then annual wellness. So we have certain benefits within the policy. Okay, this is the base one, or the what we will say the minimum plan that we have. Okay, it's called essential. So as you can see, we have an outpatient limit, we have an inpatient limit, we have medication covered within the outpatient limit. So for this particular plan, you are entitled to essential facility. We've grouped the various facilities according to the plans. Okay, I is catered for. So this is the subsidy for your lens and frame. It's 500 Ghana cedis. So I wouldn't talk about that amount, but I'll let you know what goes into the benefits. So chronic care is covered. Diagnosis, dental is also covered. Okay. Right. So general ward is covered. We do surgery and then also maternity care. So normal delivery, CS, and then assisted. And then the annual. So if you want a detailed explanation or you really want to know much about the product, we have a stand or we have a booth outside that you can consult, and then we also make the necessary inquiries. Whoa. It's not working again. Okay, so let me move on to. It's not working. Okay, so this product, My Health, is for, as I said, is for those who will not be able to be absorbed into the corporate policy. No, because of the corporate policies and all those members will be placed on the My Health. Okay, so for those ones who wouldn't be part of the corporate policy, they can be placed on My Health. Okay. These are some of the exclusions, but I wouldn't go much into the exclusions. Okay, this one I'll also not go into. Okay, so with technology interventions, what we've done is that we provided app. So when you are enrolled on the policy, you have an app that you can use to monitor your policy. Okay, so you can see your utilization, what you've done at the point in time, what is left. You can see the hospitals that you can visit. So basically, you can see everything on the policy on the mobile app. Okay. Then the HR portal is for the HR person or the one who's going to manage the policy for the company. So they also can use to take out, add on all those things that the HR person wants to do. So this is the screen for the mobile app. Okay, so as I said, you can see your benefit, you can see the hospitals and all that you have on the policy. Whoa. Okay, we also have medical advisory service, Ask the Doctor. So what we do is that within every month, so the last Friday of every month, what we do is that we have a call-in segment. So you can call in to our toll-free number. You make your inquiry, and then there is a practitioner who will be there to answer your queries or address your medical needs. And then also that's our toll-free hotline. So we run from Monday to Saturday, and then also on Sunday. It's toll-free, so you don't have to pay for that, or you are not being charged. Telemedicine, we've also introduced telemedicine. So if you would not be able to visit the hospitals, as in person, you can also do the telemedicine. So that one too, you have to call in, and then whatever, issues that you have, you'll be attended to, or it will be addressed. We have service medicine delivery tool as well. So we have a couple of them. So what you do is that you upload your prescription, okay, and then the medication will be sent to you. So basically what we do is that we send the medication to the offices. But if you want the medication to be sent to your various houses, that's also fine. We can do that for you. We also administer webinars. So they are all forms of educating our client. Okay, for Medfield, that's for those on the chronic medicine. So they have quarterly, monthly, whatever period in which you take your medication. It can also be administered to you. So we have the in-house clinic. So basically for companies who have room that they don't want most of their clients to go out, we can also do the in-house clinic. So it's a form of hospital in the office. Okay. Okay, so one will say, why choose Nationwide? So Nationwide, we have over 20 years experience, and also we have the widest HSP network. Okay, when it comes to technology too, we also have one of the best technology interventions, and also we are customer centric. So I'll leave a quote. Health is like money. We never have a true idea of its value until we lose it. So this is George Billings' way of telling us that having medical insurance is one of the best. Okay, thank you very much for the presentation.
Come to you. Okay, thank you very much, Nationwide. If there are any questions, they have a booth at the sponsors' booths, so you can discuss with them. We are about 15 minutes behind schedule, so I think we'll go to the coffee break, and which is 15 minutes long, and I think if everyone can come back after 15 minutes and we try to catch up on the time. You know, WACCBIP, we don't like to be late. So, yeah. So I think when we come back from coffee break, I have to disrupt people a little bit because we have turbo talks before lunch, and we'd like those who are going to give turbo talks to sit in this row. So apologies to everybody who is currently sitting here. We want to arrange them so that we can move through the turbo talks quickly and not waste too much time. About 30. Yeah, so this row is fine.
Sorry.
One other thing, this is very important. If you haven't registered or signed in today, please sign in the sheet.
your time.
So this ideally here should change it.
Okay.
But you see, when you stand here, the signal is low. So you have to point it.
Okay. Okay, so that means need to be. Okay, this is the signal. Okay. And then, okay, this is the pointer. Okay. And then the arrow is okay for. Okay, for forward. Hey.
You can hear me. And then right now if you talk to me inside
Is David here? David Fadewa? Hey. Oh, it's online. Is Nora here? Hey. Then see you soon here. Okay. Nora is here. David is not here.
Maybe he's online.
Theo is not here. Peter. Now, the first one, Father Ba, is he online? He's online. Oh, okay. Yeah. So you are welcome back. We'll have 3 presenters now, after which we'll have table talks before we go for the lunch break. Our first presenter, David Fadegba, is online. David, are you online?
Yes, please.
Oh, okay.
Yes.
IT team, please, are you ready?
Yes, please, I'm ready.
Yes, please, let's see David's slides.
Please, can you see my slides?
Yes. No, not yet.
That's not it.
Oh, okay. I'm sharing, but let me stop and share. Please, can you see my slide?
Not yet.
No, we can't.
Hey.
IT, please, can you take the slide off? David, please hold the line. It looks like your slide is behind this one.
Okay.
What is on is from the sponsors, so if we can take that off. Why are they struggling?
We are still waiting.
I could check number from the IT. Where is it? Sorry, we're having a little bit of technical challenge. We'll get started soon.
Oh. Good afternoon.
What's happening? Oh.
My name is Riafe Jesse.
Yeah.
And here, standing in for a colleague whose name is actually projected on screen, Nana Kwame Asante. That is our air freight operations manager. He's unable to be here due to some other engagements. Now, you may ask, who is Better Logistics, or what is Better Logistics all about? We are your preferred logistics partner of choice. Incorporated in the year 2008.
I hope you was given. I hope you
Having a little through this. Yeah. So we're incorporated in the year 2008, and we've been in operations for the past 17 years. A wholly owned Ghanaian establishment. Better Logistics is an international freight forwarding and logistics company established in 2008 with branches in Accra, Tama, and Takrady to provide you cost-effective solutions in international trade facilitation. So, like I said, we are the logistics partner of choice. Now, somebody may ask, why logistics? In this industry, I know you do imports and exports of blood samples, clinical trials here and there, and our affiliation with South Africa-based World Courier gives us the global reach to handle all such sensitive and temperature-controlled shipments on your behalf. So this is our core service area. We handle both air and ocean freight. We do custom service brokerage, among other such industry activities. So we have a strong foothold in the movement of cold chain logistics, as I've mentioned earlier, due to our affiliation with World Courier of South Africa. We do express cargo, we handle general cargo as well, and then we do our courier services as well. Because we are members of international freight forwarder networks, we have a global reach in any part of the world. We are able to pick cargo from any part of the world, from Azerbaijan to wherever, or to Zanzibar, due to the fact that we belong to this. This is just a snapshot of freight forwarder networks around the world. Elite Courier, FOR, 5-Star, Twig, and all those people. So because we are affiliated to these organizations, we have a global reach. We are able to pick cargo from anywhere in the world and bring them down as per your demands. Sorry for the technical issues. Yes, so this is also another snapshot of our area of expertise when it comes to temperature-controlled shipment and its packaging. We follow or we supply temperature-controlled packaging over the complete range of temperature regimens. -1, 96 degrees Celsius, 80, -20, +2 to 8, that is dry ice shipments, and then +20 degrees Celsius. We handle all these things on the market. So yes, through our continuous training, stringent documentation practices, and rigorous quality assurance protocols, we strive to maintain the utmost integrity in every service we provide you, from clinical trials to laboratory testing and pharmaceutical manufacturing. We maintain a robust quality management system that our clients and partners can rely on as for exceptional services while we're meeting stringent industry guidelines. So basically, we are ISO certified, ISO 9001 to 2015. And this actually demands that we run a quality management system so that we handle your goods as effectively as possible. Thank you. I don't want to take much of your time, and that is our contact as you are seeing them. Thank you very much.
Thank you very much.
Thank you.
David, are you there?
Yes, please.
Okay. Please, can we share and get him started? You have 12 minutes.
Please, can you see my slide?
No, not yet. Okay, now we can see it.
Okay.
Project.
Good morning, everyone. My name is David, and I'm a PhD fellow at WACCBIP. And today I want to talk on the topic systemic cytokine dynamics and clinical outcomes of Plasmodium falciparum. Of Plasmodium falciparum infection in Togolese children, a longitudinal study. So, malaria caused by Plasmodium parasites remains one of the dreaded global public health challenges affecting over 1 billion people worldwide. Of the 5 malaria species that infect humans, Plasmodium falciparum remains the most virulent, contributing significantly to the global disease burden with an estimated 200 million cases and 500 deaths annually. And 95% of these deaths occur in Africa, and the disease disproportionately affects children under 5 years of age and then pregnant women. The falciparum infection can result in multiple outcomes, from asymptomatic to uncomplicated or severe malaria, and ultimately to death. And ultimately to death. So what drives the infection one way or another is not fully understood, but what we know is that it involves parasite factors, host factors, and then environmental factors. And during the blood infection stage, in response to the presence of the parasite, the host immune system releases a number of pro-inflammatory cytokines, such as IL-6, NF-alpha, interferon gamma, and all these molecules play a defining role in controlling the parasite growth and elimination, thus the infection outcome. And we have also regulatory cytokines such as the transforming growth factor and also the IL-10 that maintain the balance between the pro-inflammatory and anti-inflammatory responses. And when this balance is disrupted, the exaggerated pro-inflammatory response leads to significant adverse effects that are associated with severe forms of malaria and a high mortality rate. However, this balance between these cytokines and how this balance changes over time is still not fully understood. So this study aimed to investigate the infection dynamics, cytokine profiles, and clinical outcomes that are associated with malaria in children from Togo. So this was a longitudinal cohort study, and following an ethical approval that was obtained from TBRS in Togo, 60 children were recruited following these criteria, and the children were treated with anti-malarial on start of the study, and also antiparasitics. And 15 days later, a first baseline level venous sampling was performed on each child. Then a finger prick was taken every week for up to 6 months, 3 months actively, and then 3 other months passively, with an additional venous sampling as soon as the child is positive, and 1 month later, and at the time the child develops symptoms. So this is the general workflow of the study, where we've done regular sampling and also we've processed the samples. So in establishing the outcome of the disease, the infection, so what we did is that we took total blood and then made smears, and then we went on for microscopy. And when a sample is negative, when a sample is positive, we put the sample as an early infection, and then we continue monitoring the child to see whether he will develop symptoms or not. But when a child is negative, we go on extracting the DNA, and then we do a diagnostic UPCR with the VACCBIP primers that are specific to P. falciparum, and then if positive, we continue monitoring the child until the child develops symptoms or not. And also, we've done some DBS for further analysis. We've also done blood typing and hemoglobin profile determination, and in the 60 children that we've taken, because we know the implications of this hemoglobin profile and also the blood typing in the outcome of malaria infection. And in assessing the cytokine profiles, so we've taken every samples that we've collected, and then we have plasma. And when we got the plasma, we did a sandwich ELISA, and then we assessed. That was a pilot study to assess some cytokines in the samples that we've collected. So this data was analyzed. First of all, we tested for normality, and then because the data was non-parametric, we used Kusalwali's test followed by Donet's post-hoc test. And also we did some correlation analysis between cytokine parasite densities and CT values, and the p-value of less than 0.05 was deemed to be significant. So this is the socio-demographic characteristic of my study population. So males were more represented, and the 6 years were the most predominant. And most of the children reported not to live, reported to be living under mosquito nets, and none of them was taking antimalarial or antibiotic, or they were under traditional therapy. And this is the clinical characteristics of the study population. So most of the children were O positive, and hemoglobin profile, most of them were AA, but we got some AS, hemoglobin AS, hemoglobin AC, and also one hemoglobin XS in the study cohort. Sorry. The prevalence of plasmodium infection in the study population. So out of the 60 children that were reported, we had 27 that got positive at some point, and that was 45%. And out of the 27, we had 15 that were malaria Plasmodium falciparum positive. We have 8 that was Plasmodium ovale single infection, and 3 were Plasmodium malariae single infection, and 1 co-infection, Plasmodium falciparum and Plasmodium ovale. And out of the 15 that were Plasmodium falciparum, because I was interested in these Plasmodium falciparum infections, so 8 of them were symptomatic, developed symptoms, and 7 of them remained asymptomatically infected. And we also looked at the distribution of Plasmodium infection according to blood types and then the hemoglobin profile. And we realized that most the positives that we got were O, group O, and also they were hemoglobin AA. None of the other types, variants of hemoglobin were positive among the cohorts, the children. And then for type 1 and type 2 cytokines, so we realized that symptomatic children exhibited significant elevated levels of pro-inflammatory cytokines, particularly the IL-6 and the interferon gamma, and as well as the regulatory cytokine IL-10. And this can also be explained, the levels of IL-10 can be explained by a compensatory mechanism that aims to mitigate the excessive inflammation and prevent immune-mediated damage. And we went on to do some correlation analysis, and we realized that there was there was a positive correlation between IL-6 and the parasite densities. And for the baseline children, we didn't find any correlation between the cytokines and the CT values. And as for the symptomatic and the asymptomatic, for the symptomatic one, we found a positive correlation between IL-10 and the parasite densities. We found a negative correlation between the IL-10, the cytokine IL-10, and then the TP values. For asymptomatic children, we didn't find any correlation. And this positive correlation between IL-10 levels and the parasite densities in symptomatic children, that higher parasite loads might drive a stronger regulatory response. And in conclusion, for this pilot study that we've done, this study underscored the critical role of cytokine response in shaping malaria outcome, highlighting the dually immunomodulatory role of IL-10 in regulating immune responses, and single-cell RNA sequencing and PBMCs and whole genome sequencing of the parasite are the way to further decipher the host-pathogen interactions. I would like to acknowledge my supervisors and also the WACCBIP for funding through the World Bank and Katawa's lab, and also Siri and Sunil from the Sangha for the single-cell sequencing. Thank you.
Thank you very much, David. Yep, Dr. Kwashi.
Thank you, David. I have a question about when you got the infected, those infected with malaria. I was very surprised to see such a high level of P valley. Is that what you expected? Because I thought that that's usually a minor sort of like contribution to the parasite load.
Yeah, thank you. Yeah, you are correct, doctor. But unfortunately, this is what we got in the study, especially the area where we've sampled we've recruited the children. Most of the times we get a lot of Plasmodium ovale around that area. So in Togo we have, but it's only the vivac that we do not get in Togo. But as for ovale and malaria, we get them here in Togo often.
Yes, I'm just surprised that it's so high. You know, I would expect 5% or something like that.
Yeah, it is even more than that. So it's high. It's high. But initially, this study, we are just looking at Plasmodium falciparum, just that among them, because we are doing microscopy, we got some infections by Plasmodium ovale and malaria. And also, we got to confirm them with some other qPCR.
I have a question, please. And
Any
I
other
want to know
question?
about the methodology. What factors or consideration went into the choice of days for sampling collection?
Please come again, please.
You listed some number of days.
That sample is supposed to be like our baseline, like negative samples, and then from then each week we keep monitoring, doing the testing to see whether we get a positive change. So we're doing it each week.
So why these timelines?
So
that is what I want to know.
Assuming that we know the incubation period of the parasite, which is 9 to 14 days. So we assume that we cannot, if we do weekly, we cannot miss the parasite because the incubation period is 9 to 14.
Thank you very much.
Okay, thank you very much. Because of time, okay, that will be the last.
Okay. Thank you, David, for the presentation. I just have a concern about your introduction slides, where you show the figure where IL-17 or Th17 cells are regulatory cells. That I think it's not correct.
I didn't show slides of Th17. It is IL-17. It's cytokine that are assessed. I didn't talk about the cells.
No, it was in your introductory slides where you showed the Th17 on a figure as a regulatory.
No. I didn't. Okay. No, I didn't say it is. Yeah, they are here. We are mentioning that. You see here we have T-regs and then T17. But I was talking mainly about the T-reg, IL-10, and then the transforming growth factor beta. That's the two ones that I've talked about. I know IL-17 is a dual cytokine which can be either pro or anti. It's not well defined.
Okay, thank you very much, David. We'll next have Nora.
Good afternoon. I'm Nora Owusu-Aweto, and a WACCBIP. So several interventions have been made to combat cancer in general. We have immunotherapy, radiation therapy, targeted therapy, and so on as listed on this slide here. How, in our local context, we have three main standard treatments. We have chemotherapy, radiotherapy, and surgery, of which my work focuses on chemotherapy, specifically. When it comes to how chemotherapy is impacting immune system in cancer patients, there have been several notions. Generally, chemotherapy is known to lead to a compromised immune system. However, emerging studies have shown that chemotherapy may be impacting the immune system in such a way that it helps the immune system better fight the cancer cell. I should put, okay. Oh, okay. I see. So I hypothesize that chemotherapy enhances anti-tumor immune responses in Ghanaian breast cancer patients, and these responses vary across the different subtypes of breast cancer. So the aim of my study was to determine the effect of chemotherapy on immune profiles of Ghanaian breast cancer patients, and compare these profiles between distinct breast cancer subtypes. Specifically, I aim to determine the pattern of distribution and heterogeneity of breast cancer in Ghanaian patients, to determine the effect of chemotherapy on population of specific immune cell subsets, and also to determine the expression levels of anti-tumorigenic and pro-tumorigenic cytokines. This was the general workflow of my study. I took ethical clearance from ECBAS and Kolebu IRB, and my target group were breast cancer patients who were receiving andramycin cyclophosphamide. These drugs interrupt the division of DNA during the cell cycles. They intercalate the division of DNA, sorry. And I used an analytical cross-sectional study I took samples at one time point. So for my first objectives, I extracted whole blood from three main groups of individuals. First, breast cancer patients who were receiving chemotherapy, and two control groups which are breast cancer patients who had not started chemotherapy yet and healthy individuals. So I retrieved a total of 75 samples, 39 belonging to patients who were on treatment, 33 who were breast cancer patients yet to receive chemotherapy, and 3 healthy individuals. I grouped my patients into different age group brackets, and most of my patients were between 30 to 49 years, which was consistent to several other studies, especially the WACCBIP et al. Are shown here. So I mentioned that breast cancers are grouped into either stages or subtypes. I first determined the frequency of stages, the different stages in breast cancer patients, and I noticed that most of my participants presented at stage 3 of the breast cancer, which is a locally advanced stage. One of the advanced stages is stage 3 and stage 4. Then I went ahead to determine the different subtypes, the frequency of the different subtypes of breast cancer. We have 5 main subtypes listed here: luminal A and luminal B, which are breast cancer patients that express estrogen receptor. They may or may not express progesterone receptor and do not express human epidermal growth factor 2. We have the luminal HER2. However, the luminal B is more aggressive than the luminal A. And we have the luminal HER2 type, which they have similar characteristics between the luminal A and luminal B, but they express the HER2 protein. And we have HER2-enriched breast cancers, patients who do not express either estrogen or progesterone, but have HER2 protein. And then lastly, we have the triple negative breast cancers, which are individuals, as the name suggests, do not express any of the 3 receptors. So most of my participants were triple negative breast cancer patients, the most aggressive type of breast cancer. So for my objective 1B, where I was determining the differences in populations of immune cell subsets, I used flow cytometry analysis. First, stored my cryopreserved cells and stained them with specific antibodies, and then acquired with a fax machine. This was my gating procedure to distinguish between B cells, cytotoxic T cells, NK cells, myeloid-derived suppressor cells, and monocytic antigen-presenting cells. So for the first objective 1B, I compared between 2 groups of breast cancer patients. I had on the left here, breast cancer patients who are chemosensitive, who are on treatment. On the right are breast cancer patients who were yet to receive chemotherapy. So I was checking the average median count between these 2 populations. Then I noticed that among all the immune cell counts here, cytotoxic T cells and B cells were the most abundant. However, chemotherapy did not have any significant impact on the counts of immune cells. So since chemotherapy did not impact the count of immune cells, I went ahead to see if subtype had a significant impact on the count of these immune cells. So I grouped my patients into 3 main subtypes. I grouped all luminal subtypes together, HER2 subtype, and then the triple negative breast cancers. I checked the differences between T cells, B cells, and then the other cells I listed before. I noticed that when it comes to cytotoxic T cells, between HER2 patients and triple negative breast cancer, HER2 had high expression levels, the highest expression level of CD8 as compared to the other cells. And we had between the 2, HER2 and triple negative breast cancer, HER2 expression count was significantly, HER2 had a significant expression level of CD8, which means that in HER2 patients, they may be responding better to the chemotherapy as compared to the other subtypes. When it comes to B cells, chemotherapy did not have any significant impact on them. Then I checked the other immune cells, NK cells, myeloid-derived suppressor cells, and subtype did not have any significant impact on these cells. So for my last, So for the summary of my objective 1, I noticed that chemotherapy does not have any significant impact on immune cell count, and then cytotoxic T cells had a significant impact. Cytotoxic T cells were significantly higher in HER2 patients as compared to the triple negative breast cancer. And then for the other immune cells, there was no significant difference across the subtypes. So as I mentioned earlier, elevated T cells in HER2 patients indicates that HER2 may be responding better to chemotherapy. And then the lack of significant shifts in the other immune cell populations indicate that chemotherapy does not strongly affect the immune system. For my last objectives, I used Luminex assay to determine the expression levels of pro-tumorigenic and anti-tumorigenic cytokines. Then I noticed that for pro-tumorigenic cytokines, IL-2, TNF-alpha, IL-10, and IL-1 beta had significant impact when I compared between chemosensitive and chemo-naive. The expressions of these pro-tumorigenic cytokines was very high in the chemosensitive patients, especially as seen in IL-2 and IL-6. Then I went ahead to check anti-tumorigenic cytokines, and I noticed that for interferon gamma and IL-2, there was some slight significance difference between chemosensitive and chemo-naive, where chemosensitive was expressing a bit higher interferon gamma and IL-2 as compared to interleukin-12 P70. So I concluded that chemotherapy induces anti-tumorigenic cytokines in immune cells. Chemotherapy induces anti-tumorigenic cytokines in patients who may be responding to their treatment. For subtype, so I went ahead again to check whether subtypes have a specific impact on IL-6.
I think you should be summing up.
Yes. I went ahead to check whether subtypes have a significant impact between cytokine expression. I noticed that subtypes did not have any significant impact between all the cytokines that I listed here. So for the summary of my objective 2, chemotherapy induces a pro-inflammatory responses which may have unintended effects of tumor progressions or recurrence. Chemotherapy may simultaneously induce anti-tumorigenic cytokines in patients who are responding, and neither pro or anti-tumorigenic cytokines showed any significant expressions across the subtypes. So for my objective 2, elevated interferon gamma and interferon IL-2 may serve as biomarkers for positive immune responses in chemosensitive patients. And then increased IL-2, TNF alpha, and IL-10 may also indicate inflammation related side of immune responses. Related side of effects of immune escape mechanisms after chemotherapy. So for the limitations of my study, I took my samples at one time point. However, there are other studies in my lab where they are looking at the longitudinal effects of chemotherapy. They are following up on the same patients to see whether there is a trend after different time points. And then I use a smaller sample size, 75 samples. Maybe this study could also be repeated with a larger sample size. And then my samples were not matched across all the groups of individuals that I was comparing.
Can we end here?
So these are my references. And
okay.
I would like to acknowledge all my supervisors and all the people listening. Thank you.
I think we have enough time just for one question. Yeah.
I'm just curious to know, since age has been related to the cancer cells, breast cancer, I just want to know if the shape of the breast, whether it is saggy, roundish, has anything to do with breast cancer. It's scientific. It's evidence. So maybe there is a connection between a saggy breast, a roundish breast, and then the breast cancer. I'm not being. I'm looking at whether it can be used as a marker, so that when you see somebody with a saggy breast, the chances of the person having a breast cancer will be. I'm curious to know.
So for now, I cannot tell if the shape of a breast can. But then there are other studies in my lab where they are comparing age variations in immune responses. So maybe after that study, we can tell for sure if the breast.
Thank you, Nora. Yusuf. Our next presenter is Yusuf. We want to remind you that you have 12 minutes.
Good afternoon. I'm Yusuf Mubarak, and I'm going to show you how a common food contaminant called ochratoxin A enhances the replication of a B virus by suppressing the type 1 interferon signaling pathway. The type 1 interferon signaling pathway is a critical component of the innate immune system, which has been found to possess antiviral
anti-cancer, and immunomodulatory properties. Because of that, recombinant interferons are employed for the treatment of hepatitis virus infections, and they are effective, even though it comes with its own adverse effects. The type 1 interferon has pathway has two major components, the upstream and the downstream. The downstream is the JAK-STAT pathway, and the upstream can be activated by viral infections, leading to the expression of interferon alpha or beta. Secreted interferon alpha or beta interacts with the interferon receptor complex that activates the JAK-STAT pathway to result in the expression of interferon-stimulated regulatory elements. So it is the interferon-stimulated regulatory elements that processes the antiviral, the anti-cancer, and then the immunomodulatory properties of the pathway when it is activated by, let's say, viral infections. And over hundreds of ISRE are expressed. An example is the OAS3, which has been shown there. But mycotoxins like aflatoxin B1 and citrulline has been shown to suppress the activation of the type 1 interferon pathway. However, ochratoxin A, which is the compound of interest, effect of ochratoxin A, which is the compound of interest, and also a mycotoxin and a major food contaminant on the type 1 interferon signaling pathway has not been established. This is the structure of ochratoxin A and is secreted by Aspergillus and Penicillium species, causing nephropathy in the kidney and also inflammation in the liver. And because several investigations have revealed that ochratoxin A can cause cancer in animals, but this has not been shown in human. It is classified as a group 2B human carcinogen. It is chemically stable at boiling temperature, indicating that just boiling your food will not degrade ochratoxin A. So you need to heat it at higher temperatures, like above 180 degrees. And it has a long serum half-life in humans. Ochratoxin A contaminates cereals, vegetables, and even processed beverages. In a study that was conducted in Ghana that analyzed the levels of ochratoxin A in maize sampled in 6 regions were found to be contaminated with high levels of ochratoxin A. In fact, 49% of maize sampled in these regions had the levels of ochratoxin A high above the Ghana Standard Authorities levels. Other African countries like Nigeria has also reported high levels of ochratoxin A in foods, and even European countries are not spared with ochratoxin A contaminated foods. So because of its global occurrence and its effects on the immune system being an immune suppressor, we aim to determine the effect of ochratoxin A on the type 1 interferon signaling pathway, which has been shown to have antiviral, anti-cancer, and immunomodulatory properties. So we also assess its effects on the expression of key signaling transcripts in the pathway, and we further model the effect of ochratoxin A on the replication of Hep B virus. So specifically, we've determined the inhibitory effect of ochratoxin A on the type 1 interferon signaling pathway. We've also determined the effect of ochratoxin A on the expression of key transcripts in the pathway, and we further determine its effect on the replication of Hep B virus. So 3 cell lines were employed in this study: the HEC293, the HEPG2, and the HEPG2 2.215. And to establish these objectives, first we determine the maximum non-toxic concentrations of ochratoxin A. By doing that, we seeded 40,000 cells. Then the cells were treated with different concentrations of ochratoxin A, and we performed the MTT assay. And here the results reveal that 4 micromolar of ochratoxin A
and was able to yield a percentage variability which was greater than 80%. So that was selected as the maximum toxic concentration. So this shows that indicates that in the subsequent experiment, the maximum concentration of procatoxin A that we can use to treat the cells is 4 micromolar. We also determined the minimum concentrations of procatoxin A that will maximally activate the JAK-STAT pathway, because you need to activate or stimulate the pathway before you start with the treatment. And to do that, we use the dual luciferase assay. In the dual luciferase assay, 2 plasmids were used. The PISRE, that expresses the firefly luciferase, serving as the experimental reporter, and then SV40, that expresses the renilla luciferase, serving as the internal control reporter to normalize the experimental reporter. So in the setup, the 2 plasmids were transfected into the cells, and here we expect our reporter proteins to be expressed simultaneously when the pathway is activated. So this is how the setup was. This is how the setup was. The transfected cells were treated with different concentrations of interferon alpha, and we performed the MTT assay. And here, 400 international units per ml was able to maximally activate the JAK-STAT pathway in all the cell lines. So this also indicates that the maximum concentrations of recombinant interferon alpha that will be used to stimulate the cell lines before they are treated with procatoxin A is the 400 international units per ml in the subsequent assays. So this takes us to the specific objective 1, which is to determine the effect of procatoxin A on the type 1 interferon signaling pathway. And to do that, the transfected cells were treated with procatoxin A or interferon alpha. Then we performed the dual luciferase assay. And here the results reveal that as compared to cells that were stimulated with only interferon alpha, procatoxin A was able to, cells that were treated with only interferon alpha, that maximally activated the pathway. Procatoxin A treatment was able to significantly suppress the activation of the pathway in a dose-dependent manner in all the cell lines. Okay. So having been able to determine that, we went on to see which transcripts are affected. So specifically, we analyzed the tyrosine kinase 2, STAT1, RF9, and OAS3. And to do that, the and to do that, the HepG2 2.2 alpha cells were treated with procatoxin A and interferon alpha, and RNA was extracted, cDNA was synthesized, then QRCT-PCR was performed. Here, the results reveal that compared to cells that were stimulated with only interferon alpha, procatoxin A was able to significantly suppress the expression of key transcripts, STAT1, tyrosine kinase 2, AS3, JAK1, RF9 in a dose-dependent manner, as observed in the in the JAK-STAT pathway. So this confirms the ability of other mycotoxins to significantly suppress the activation of type 1 interferon pathway. So having been able to do that, because the type 1 interferon pathway because the type 1 interferon pathway has been shown to suppress the replication of a B virus, and we've shown that treated with only interferon alpha significantly suppress the replication of B virus DNA. But cells treated with rocatozin A and then a recombinant interferon significantly promoted the replication of a B virus, indicating that rocatozin A has the ability to promote a replication virus in Hep G2 2.215. Okay, so here we've been able to show that rocatozin A suppresses the type 1 interferon pathway, also decreases the expression of key transcripts in the pathway, and further promotes the replication of a B virus. So the take-home message here is that a food contaminant, a common food contaminant called rocatozin A, promotes the replication of a B virus by suppressing the type 1 interferon pathway via the JAK-STAT azoles. So special acknowledgement goes to my supervisors, and then the lab team, WACCBIP, and then KNUST for this opportunity. Thank you.
Questions, please. At the back.
Thank you very much. That's quite interesting. Simple question: How does it stimulate the STAT2 pathway? For example, is it a transcriptional activator? And if it is, of what specific genes in the pathway? I'm asking if how does ochratoxin A stimulate the pathway that you described? Is it as a transcriptional activator, or does it bind any protein that increases the transcriptional activity within the pathway?
Okay, so the mechanism, so in this experiment, the pathway was activated using the recombinant interferons. But how ochratoxin A affects the pathway, whether it binds to specific proteins, has not been determined. So it's not yet known whether they bind to these proteins or not. That has not been determined.
So what warning do you have for us? I mean, because many of us, we love to, particularly at a season like this, take maize.
Okay, so for example, in coffee production, what they do is they prolong the roasting of coffee. So you may have to increase the heating of your food, but it also has its effect.
Increase the heat?
No, yes, like prolong the heating, the boiling time. Yes.
I thought you were indicating that boiling may not affect
I said okay, so the temperature, yes. So in high temperature periods, around above 180, is able to degrade.
Degrees centigrade?
Degrade, yes. 7 to 90. Even that is 7 to 90. So it's a stubborn food contaminant. Even processed beverages, wine and the rest, there is some sort of ochratoxin A in there. Yeah.
Okay. Thank you very much. So we are done with the presentations. We now want to invite Dr. Abiola to take over and run us through the table talks.
Please, let's accommodate the chairs. All right, good afternoon, everyone. I'm confident that we are having an awesome time enjoying the conference. This afternoon, we have our wonderful fellows here to talk to us about the work they have been doing for some time. So they will be inciting us about why you have to pass by their poster stand to get to know how important and relevant their studies are. So one after the other, they are going to come on stage and they are going to talk to us. In 2 minutes, one slide, no animation. No video. The moment is 2 minutes, I'm going to ask you to step down. Please, we are far behind schedule, so make it snappy. So one after the other, we are starting with Mata Akle Asare.
Hello, good afternoon. My name is Martha Asari, and I'd like to talk to you about antimicrobial resistance. Antimicrobial resistance continues to be a threat to our very existence. As when patients are placed in the hospitals, they are essentially, when clinical patients are being placed in the ICU, they are exposed to many bacteria, especially those highly resistant to antibiotics, even those of last resort. And those antibiotics are inclusive of carbapenems and colestins. And it has been found in a previous study by my supervisor, Dr. Abiola, that these pathogens are highly resistant to these pathogens, which were isolated from the ICUs in Ghanaian hospitals, were found to be resistant to last resort antibiotics. And as a result, we need to look at alternative therapies. And these alternative therapies could include adjuvant, combination therapy, and immune stimulation options. And obviously, I'd like to talk to you about HSD 1624. HSD 1624 was synthesized by my co-supervisor, Prof. Herman Osintim in the United States, and this compound was found to be a small molecule of antibacterial activity and was able to potentiate colistin in foreign isolates. And it had its mode of action being memory disruption and redox, or the production of oxidation species. And to be fully able to understand it and use it, or for it to be eventually approved onto the market, it will have to be fully profiled, and this includes looking at its anti-biofilm activity and biofilm eradication activity, its influence on the...
Time up, Martha, thank you.
I would like to invite you to my poster number 1.
All right, Martha, thank you. It's okay to be nervous. All right, so don't worry about that. Feel at home, okay? And second person, number 2.
Good afternoon, everyone. My name is Eugene Lamte, and I'll be presenting on Plasmopred, which is a machine learning-based web application for predicting small molecule inhibitors of the apical membrane antigen 1 rupture net 2 protein invasion complex. So, at the blast stage of malaria, the AMA1 and RON2 interaction leads to the formation of tight junction, which ultimately leads to the Plasmodium merozoites invading the red blood cells. So for our project, we are looking at finding small molecules that can actually block this interaction and prevent the invasion of Plasmodium merozoites into the red blood cells. So to do this, we implemented the machine learning pipeline, where we got data from PubChem, which is over 360,000 compounds, which are inhibitors and non-inhibitors of the AMA1-RON2 interaction. We then pre-processed this data, and then the pre-processed data was used for our model training, our model evaluation, and model validation. We then took the 3 best models and implemented it in our web application, which is Plasmopred. Currently, Plasmopred is online and is running, so anyone interested in Plasmopred should check out Poster 2. Thank you.
Thank you, Eugene, for keeping to the time. Enos.
My name is Enos Kwashi, and then I'm presenting on the need for happy surveillance and natural substitutes for chemical insecticides. So the use of synthetic insecticide is one of the most commonly used malaria control method or mosquito control method in Ghana, especially in the southern regions. Whereas people in the north, most of them, especially those in the rural and deprived communities, do not have access to this due to the hefty price tags that comes with it. So, but beyond the affordability, these insecticides, synthetic insecticides, also comes with some environmental and health threat. They also lead to the creating of pesticide treadmill, where the mosquitoes become resistant due to overuse of these chemicals. So most people in the rural communities in the north, they rely on a plant called herpes surveillance, called in Dagbani Dumsinamini mosquito rival. A plant, it's an aromatic plant that is known for or claimed by these people to repel mosquito. So usually they just pluck part of it or burn the dry one in the room just to ward off these mosquitoes. So, yeah.
All right, thank you. Poster number 3. No, poster number 3.
Yeah, poster number 3. Kindly visit.
Did you know that plasmids are implicated in the spread of antibiotic resistance? My name is Gloria Adu, and the title of my poster is Cure the Plasmid, Kill the Pathogen. So it's about how to target plasmids and reduce the spread of antibiotic resistance. So if you want to find out how plasmids can be cured and how that will affect their response to antibiotic treatment, meet me at poster 5. Thank you.
Yeah. Thank you for saving us some time.
All right, thank you very much. I want to share a brief insight of what we are also doing in the north. So imagine you are being rushed to the nearby health facility where empirical treatment decision could mean the difference between life and death. What will be your fate? So this is the context that shaped this study. So I'll be inviting you to come to poster number 6. Thank you.
Thank you.
My name is Charles Robertson, and my poster is about development of high glucose levels and high uric acid levels after consuming a lot of clowns and for a long time. So I gave clowns to rats. I used rats as my test models, and after 60 days, that's about 2 months, I noticed that there were high glucose levels and high uric acid levels. That is, even though clowns have omega-3 and are very good for their nutritional benefits and other health benefits, there's a likelihood of getting certain metabolic diseases. I am at poster 7. Thank you.
Thank you. Albert.
Hello everyone, my name is Albert Boahen and I'll be presenting on evaluating the antimalarial potency of VA22 against different Plasmodium falciparum strains. So the emergence of drug-resistant Plasmodium parasites poses a significant threat on the eradication of malaria. This highlights the urgent need for new therapies targeting different mechanisms of actions to combat the disease. To accelerate this, drug repurposing and repositioning offers a better alternative by discovering new uses for already existing drugs. In my study, VA22, an anti-cancer agent in the second clinical trial phase, and its structural analog, NS245, were screened against some Plasmodium falciparum strains. To know more about my work and how it went, you can visit poster number 8. Thank you.
Awesome. Thank you, Albert. Virginia.
Good day, everyone. My name is Eugenia Emma, and I'm here to introduce my research titled Doxorubicin and P-coumaric acid co-treatment for prostate cancer cells. Prostate cancer is known to be one of the leading causes of cancer-related death among men. It is ranked as the 5th globally and then the 2nd in sub-Saharan Africa. Our research made use of PC3, prostate cancer cell lines, that are known to be very aggressive. They are highly metastatic, and then they lack the androgen receptor, which makes the use of hormonal therapy ineffective in managing this subtype of prostate cancer. Currently, doxorubicin is used as a standard treatment for prostate cancer. Despite it being established to be a potent anti-cancer agent, it has several limitations that include ability to induce cardiotoxicity and then development of drug resistance. Research is ongoing to explore the use of natural products as an anti-cancer agent, and then P-coumaric acid has been shown to be highly effective in inhibiting cancer, that in having anti-cancer properties such as anti-metastatic potential and then induction of apoptosis. And so despite both drugs being established as a good anti-cancer agent, the use of it in combination therapy has been underexplored. And so our research aimed to investigate the therapeutic effect of combining doxorubicin and P-coumaric acid on prostate cancer cells. If you are curious about what we did and then what we found, please join me at poster number 9 for further details.
Thank you, Eugenia.
Yeah, so I'm Francis. I work with Dr. Lucas Amenga Etego on the project on spatial epidemiology of malaria. I should be able to present to you some data from our study so far on urban malaria. And largely, we seek to investigate the spatial temporal dimension of urban malaria, looking at the prevalence and the seasonality of these data sets. And largely, we used the Bayesian inference, and that includes INLA, the famous INLA integrated nested Laplace approximation, and stochastic methods to explore this data and get deeper into it. Please do come to poster number 11 so we can digest this. I will not bore you with the mathematics behind this because it can be very intimidating. So please do feel free to come.
All right.
I'll share with you some visualizations and then we discuss further. Thank you very much.
Thank you, Sir Francis. Something else to digest after lunch. Mona?
Good afternoon. I'm Mona Lisa Ibieresechi, and I'm here to invite you to my poster presentation this afternoon on nationwide surveillance of molecular markers for anti-malaria resistance in Ghana. just to borrow Minister's words from yesterday, the mosquitoes do not need passports to travel. So does the parasites that they carry. And so this migration introduces new and existing resistant markers to the communities, and hence the need for continuous nationwide surveillance for these mutations that have been transported, hence to inform policymakers on the latest trends in it. So not to bore you, I would like to invite you, to my poster this afternoon to hear more about the findings that have been generated from the study. Thank you very much.
Poster number 12. Poster number 12. Yeah, Theophilus.
So good afternoon to you all. I'm Theophilus Pinto, and my research is on food safety. So that's edible offal. So with what the introduction of One Health, scientists are now putting in effort to investigate all forms of how resistance bacteria come about and how it is spread. So I decided to investigate offal sample because offal is a delicacy here in Ghana, and I believe you can see from the picture we have some edible offals from here. So we aimed at investigating ESBL producing E. coli. So I would kindly entreat all of you to join me at poster number 14 for a detailed information. Thank you.
Theophilus, I will come. I like fufu and light soup.
Good afternoon. I'm Nunana. I'm a bioinformatician here at WACCBIP. So anti-malaria drug resistance remain a threat to malaria control and elimination in Ghana. So imagine the parasite, which is Plasmodium falciparum, developing resistance to our first line anti-malaria drugs, that's ACT and sulfadoxine pyrimethamine for intermittent preventive treatment in pregnant women. This could lead to serious consequences such as loss of lives, as well as drawing us back in terms of our efforts that has been made. So we monitor the prevalence of P. falciparum mutation linked to resistance in this drug. So back in 2005, Ghana said, chloroquine, you are fired. This led to the continuous nationwide rollout of artemisinin for malaria prevention. So in our study, we reported WHO validated markers as well as mutations associated with partial artemisinin resistance. Please come to poster number 15 and let's discuss more. We have to fire malaria.
Yes.
Say fire.
Yes, fire. Thank you.
Hi, good afternoon. My name is Stephanie. In the spirit of firing malaria, the Muzi Biome Lab is looking at mosquito gut microbiome and their interactions with Plasmodium falciparum. The reason we're doing this is because we want to find out if there are bacteria within the midgut of the mosquito that's eliciting any toxic effects on Plasmodium, and we're hoping to use this to develop paratransgenic approaches that would kill the parasites within the mosquito before it's able to transmit it to the next person. I have exciting results to share if you are happy to see how mosquitoes and their bacteria within them are bringing us closer to the end of malaria. Please pass by poster number 16 for more information.
Thank you. Oh.
Good afternoon. My name is Gloria Menu, and I'll be presenting a poster on the topic Interacting Species, a study of co-infections and host responses to SARS-CoV-2 infection. Have you ever wondered what really happens when multiple infections invade the host? This is what my study is trying to investigate. Our results are quite interesting. we've observed some patterns and also some hints of interaction. And the data is not all about matching the bacteria or the pathogens and then the hosts, but also the story that microbes tell when they show up together. So if you are interested in what they whispered or shouted, come to poster number 17 to listen to more.
Thank you. Nice.
Hello everyone, good afternoon. I'm Seraphim and I'll take you through the growth dynamics of Serratia mastitis, a bacteria we've been exploring in the Multibiome lab. It's in the mid-gut of the Anopheles gamma mosquito and its implications for malaria control. So the Serratia mastitis is found in the mid-gut of the mosquito and is known to have antiplasmodial effects. In studies that have highlighted its antiplasmodial potential, you'd find that they would first of all introduce antibiotics to the mosquitoes to clear majority of the mid-gut microbiota, after which they would incorporate their bacteria of interest and then provide an infectious blood meal to assess the effect of the bacteria on the plasmodium. So we want to ask the question, what happens to Serratia if we leave it to behave naturally? No antibiotics and no boost. And what is the effect of different concentrations of Serratia on the influence of Serratia proliferation post-blood meal and the effect on the Plasmodium parasite? So in recent studies, they find that Serratia proliferates at 24 hours post-blood meal. But for our studies, because we left the bacteria intact, we had it at an early proliferation window at the 12-hour time point post-blood meal. So we further went on to introduce the different concentrations of Serratia.
You had one slide. You are
Okay,
telling us
please
about
come
the
to
work
poster number 18. Thank you very much.
Thank you. Yeah, poster number 18. Daniel.
Hello. I'm Daniel, and then I'll be presenting a poster on HIV infection-associated leaky gut as a risk factor for pernicious anemia in HIV patients. So when we ask most of us here what is a disease that's associated with HIV, most of us are going to say AIDS. And then that makes sense because for as long as we have known about HIV, AIDS has been the condition that has been associated with it. But then since the advent of antiretrovirals, this narrative has changed because we are seeing that the progression to AIDS has decreased, and then this has decreased the mortality significantly. Now the question goes on that, does it mean that it's game over for HIV? Unfortunately, that's not the case, because we are seeing emerging cases of non-AIDS comorbidities in HIV patients, from conditions ranging from metabolic and cardiovascular diseases. We are seeing neurocognitive disorders, and then also we are seeing autoimmune diseases. This goes to show that there is still much work needed to be done. So in my study, I'm looking at leaky gut, which is rapidly gaining attention in HIV patients, and then how this condition leads to an autoimmune disease. Thank you. If you are interested in knowing what I did and then the results I found, you can join me at poster number 19.
Thank you, Daniel. Poster number 19. You're precious. 2 minutes.
Good afternoon. I'm Precious Kumi. Booster vaccinations are necessary to sustain immunity against SARS-CoV-2. However, how different booster platforms or vaccines combine to shape the durability, the magnitude, and then the breadth of immunity, especially in an HLA diverse setting such as ours, remains unclear. Our study evaluated the magnitude and then the durability of humoral and cellular immune responses against 2 COVID-19 vaccines that had been administered as boosters. We found platform-dependent patterns that I would like to share with you. So kindly come to poster number 20. Thank you.
All right, thank you, Precious. Poster number 20. Caleb. 2 minutes.
Good afternoon. My name is Caleb Owusu. So currently, there are only 2 WHO approved malaria vaccines, the RTSS and the R21. However, both vaccines are limited in their efficacy and the duration of the immunity that they provide. As a result of this, many labs and researchers across the world, including here at WACCBIP, have come up with or proposed various vaccine candidates that can potentially provide the long-lasting immunity needed to combat the disease. 2 of these vaccine candidates identified here at WACCBIP are the PF merozoite-associated armadillo-protein and the PF asparagine-rich merozoite antigen. Serological studies have shown that antibodies against these proteins prevent or inhibit merozoites from invading red blood cells. They are also shown to be associated with reduced risk and also complete protection against malaria. But these studies alone are not enough to justify their therapeutic potential. One very key determining factor of their suitability as vaccine candidates is their essentiality to the growth and survival of the parasites. So for my work, I was able to tag the genes that code for these proteins with a Glimex ribozyme and an HA tag, which enabled me to knock down the expression of these proteins separately in order to assess their relevance to the growth and survival of the parasites. For more on this, see me at poster number 21. Thank you.
Thank you. Poster number 21. Anobi.
Good afternoon, everyone. My name is Joseph Into Honorable University of Ghana MPhil student from Kamara Lab, and my poster is focused on long-term immune response induced by hepatitis B virus vaccine. Hepatitis B virus is a major global health threat which can cause liver cancer or cirrhosis. We have vaccination interventions coming to play to help provide long-term protection against this disease. Now, the WHO recommended HBV vaccine is a 3-dose schedule, which usually may be cost-effective and also may be challenging in getting participants back for all the 3 doses. So, looking at factors such as diverse genetic backgrounds, co-infections here in Africa, and also other environmental factors, the question remains: do we all need the 3-dose schedule for this HBV vaccine for long-term protection? Can fewer doses provide long-term protection? And to answer this, our experiment seeks to investigate how 1, 2, or 3 doses of the HBV vaccine influences the kinetic and quality of long-term immunity. And if you want to find more about the study, kindly visit poster number 22, and trust me, you will find interesting findings that can change WHO recommendations.
Poster number 22. Anobi, thank you. Sandra.
Hello. The fight against antibiotic resistance is still ongoing. However, nature has provided us with a solution to this problem. In my lab, that's the MOSI lab, we've been able to isolate a lot of bacteriophages with activity against mycobacterial infections. To further understand how these natural bacteria enemies cause problem or kill bacteria, kindly pass by poster number 23. Thank you. My name is Sandra Benny.
Thank you. Poster number 23. Comfort.
What comes in mind when you think of the mango fruit having therapeutic potential against prostate cancer? My name is Comfort Adamesi, and in this afternoon, I'll be presenting on an in vitro investigation of the anti-prostate properties of the different parts of the mango fruits. We have exciting results for you. So please, if you want to know more about this, we know the men are suffering, and this is something which is actually going to be of great help to you, because treatment against prostate cancer is expensive. So if you want to know more about this, please visit poster number 25. Thank you.
All right, for mango lovers, poster number 25. She said men are suffering. Elizabeth.
Hi everyone, good afternoon. I'm Elizabeth Bugasi from Commensalism to Resistance, Iron Metabolism and Adaptation Enterobacter Chlorwacke Complex. I'm here to tell you why you can't leave this conference without visiting my poster. Of course. So, Enterobacter Chlorwacke Complex, please the next slide. Why am I so interested in the species that belong to this complex? So they are usually part of the gut commensals where they are harmless to humans, and they are usually found in the colon where they feed on undigested carbohydrates or proteins. However, under certain conditions, they are able to transition from being harmless gut commensals to being pathogenic bacteria. And hold on, that's not the catch. The catch is that bacterial species that belong to this complex have the ability to resist multiple antibiotics. So when they transition from being commensals to pathogens, they actually cause very difficult to treat diseases, which is why I'm interested in them. So my study focused on iron as a way of understanding the factors that are facilitating the transition from being commensals to pathogens and to resistance. And iron, because iron is a very essential micronutrient. It's required by nearly all forms of life. I bet you really don't want to miss all my findings. Come, let's have a great discussion at poster number 26.
Thank you. Poster number 26. Flavia.
Good afternoon, everyone. My name is Flavia. Please move to the next slide for me. I don't know if you remember the last time you had fever, but this is different for children, especially those who cannot really express what is wrong with them. And at the time when all fever cases were being treated as malaria, it was kind of easy for the clinicians. But now we've been able to tell that there are lots of other things that cause fever, a whole lot of pathogens, and this poses a very significant threat to diagnosis, especially the fight against AMR. And so in the hospital settings, when someone is negative for malaria, you're always left with the option of either treating with antibiotics or a whole lot of other pathogens that you have to check for. And the main challenges are also that even if you have a single test that is able to detect most of these pathogens, you don't know if that's actually the cause of infection. And so we are trying to move separate from the pathogen detection to host responses.
Okay, so it was a cross-sectional study, and then I used about 165 female sex workers who gave me their high vaginal swab sample, from which I tested for the presence of the Candida species using the Alplex candidiasis assay in the multiplex PCR. Okay, so my findings indicated a high prevalence of about 54.29% in the population, with the species differentiation as shown, with Candidans being the highest, showing 65%, and then parapsilosis showing the lowest. This shows that the presence of non-albicans species, which are not common or involved in these infections. So this gave us that this showed us that non-albicans species are also involved in Candida infections, and then they should also be targeted interventions to prevent the spread of them. Thank you. If you're interested in knowing more about this, then visit my poster. That's poster number 28.
Poster number 28, Emmanuel. Bright.
Hello everyone, good afternoon. My name is Baniel Bright, and I'm here to tell you about ISKMV, a pathogen that is fast expanding its territory. I believe we all here love our roasted tilapia and our roasted catfishes, but there's a pathogen called ISKMV that is causing havoc in these fish farms or in this territory of organisms. ISKMV is, in 2019, ISKMV had an outbreak in various tilapia fish farms along the Lake Volta, where it led to massive death and huge economic losses. This particular outbreak was later confirmed to be caused by the infectious spleen and kidney necrosis virus, ISKMV, which was the first ever recorded in Ghana and in Africa. The need for a genomic surveillance system has become very evident since the host range of this particular pathogen is very broad. And so My study was designed to monitor the transmission and evolution of ISKMV in catfish, which is a novel host, using this as a robust genomic surveillance system. This study requires both molecular identification techniques as well as in silico analysis. To know more about my valuable insight into ISKMV, visit poster 29. Thank you very much.
Yeah, poster 29. Catfish for free. Michael.
Good afternoon. My name is Michael Brown, and we did an interesting study on knowing whether Fusobacterium, which is implicated in dental plaques, can cause cancer. And we know now, as of 2024, that Fusobacterium nucleatum is implicated in colorectal cancer. Come to poster 30 and find out if your dental plaque can cause cancer. Thank you.
Thank you so much, Michael. I hope you agree with me that our students, they are doing a very, very great work. Don't you think so? So if you think so, can you please put your hands together for them? So I'm going to hand over to Dr. Kwashi. Thank you. So if you think so, can you please put your hands together for them?
Yes, for who gives talks versus who gives posters is very tough. And I think to really get a full breadth of what's happening also within Ghana, it's very good to visit the posters, interrogate them, ask them to basically questions. And I think I'm hoping that while you're seated here, especially for the international visitors, you're getting ideas. Maybe you can see projects where there's room for collaboration. You know, one of the points of this meeting is collaboration. Right after here, we're going to have lunch. We'll have the poster session. And then, so that's 2-hour time where while you're looking at the posters, you're discussing, oh, I heard your talk, I think that's interesting, maybe there's ways we can collaborate. And then, right after lunch, we'll come back for the panel discussion, which I think will be quite interesting. Every year, we try to make the panel discussion as relevant as possible and as future-looking as possible. So, I think these guys are ready. I don't see anybody. Oh, yes, DCL. So, I think it's just going to be 5 minutes, and because we have 15 minutes, so I hope you bear with us.
Thank you, Dr. Kwashi. Good afternoon, everyone. My name is Nanesi Hansen, and I'm with DCL Laboratory Products Limited. Many of you are familiar with us. We are a medical supply company based here in Accra, and we've worked with many of you. All right, so I just wanted to take some time to go over some of our services. So we are a channel partner for many original equipment manufacturers of life science research and IVD devices. And we not only supply, but we actually provide after-sale support as well. So when you purchase from us, know that throughout the lifetime of the instruments or kits, we're here for you in terms of providing support, in terms of maintenance and everything else. We're going to be with you in that process. Right. So I just want to talk about, so like I said, we provide clinical solutions, hospital solutions, and research and academia solutions. However, because of the setting, I'm going to focus on our research and academia solutions. All right. So we've partnered with several original equipment manufacturers, including QIAGEN. QIAGEN is the major one, right? For everybody here does some molecular work. one way or the other. So QIAGEN is one of our major partners, and we're with them through the entire PCR workflow. So I'll go through that in the next slide. We're also partnered with MEG. So for your chemicals, your benchtop equipment, anything filtration, we're here for you. In terms of microbiology, whether it's your media, your AST discs, I know some of you are working with pathogens and you may want to do some culture and sensitivity there. We also work with Thermo Fisher to supply their Oxoid products. We're partnered with Zeiss, and we actually have some wonderful microscopes at our booth, so you can come by and take a look at them and see if they will fit your needs. I know some of you have already been by, but you can come by and take a look at that. An area that most people are not aware of that we are involved in is histopathology. So we're partnered with Sleep, which is a German company for histopathology. So everything from your tissue processing through sectioning, or embedding, sectioning, cover slipping, everything, whether it's consumables or equipment for histopathology, we have solutions for that. So if that's something you're interested in, we're not exhibiting anything, but you can talk to me about it if that's something you're interested in. So I just wanted to spend some time focusing on QIAGEN, because I know that that is what we're actually going to do. It's relevant to most of you here. So QIAGEN provides solutions for the entire PCR workflow, as well as other molecular applications. So we have options for sample disruption, such as our tissue ELISA. For sample purification, we have different manual and automated products. And then we have the QIAxcel Connect for quality control. So for those of you doing NGS or any sort of need capillary electrophoresis for any applications, there's a QIAxcel Connect. It will do your RNA quality analysis. It has applications for fragment analysis, whether HLA haplotyping and other things. We also have options for a small instrument for your assay setup. Yes, so then we also have the kits, and a lot of people don't know, we have instrumentation and kits for real-time PCR, and we have a wonderful integrated platform for digital PCR as well. So I'd love to talk to you about that if that's something you're interested in, as well as solutions for next-gen sequencing. QIAGEN doesn't have a platform for it, but we have kits and we have software. So I know my time is limited today, but there's a lot of solutions that DCL offers, and I'd love to talk to you about that. So come by our booth. We're with all the exhibitors, and I'd love to meet the rest of you. Thank you.
Thank you, Nanesi. So I think, is there one more? Yes, okay. There's one more. One thing I also forgot to mention is that after the panel discussion, sometimes we design the schedule deliberately to force everybody to be engaged during the course of the day. The panel discussion sort of breaks up the post-lunch fatigue, because it's quite engaging. And then we have 3 very excellent plenary talks that will close out the day. So I thought I'd remind you about that. So final sponsor presentation, and then we are free to go for lunch. And just like yesterday, faculty, plenary speakers, and invited guests will be on my right when we go out. I think it will still be on the right, and yeah.
Hello, good afternoon. So my name is Jora from Incaba Biotech. So today I'm going to talk to you on some of our core services. Okay. So Incaba Biotech, we are originated from South Africa, but we have branches all over Africa. And today, this year, West African region, we are celebrating 10 years. So we are going to Nigeria for the 10th year anniversary. So what are we into? So the main aim of Incaba is to collaborate and also to promote molecular techniques among researchers across the continent. So what we do is that we support researchers, we organize or support like conferences like WACCBIP, and also when it comes to institutions, whenever they have conferences, we organize workshops as well to, as a way of promoting molecular techniques across the country, across the continent. Okay, so what are some of our core services? So our core services are to manufacture DNA synthesis. So when it comes to your probes, your primers, we synthesize that, and within 2 weeks you get your oligos to work with. And also when it comes to our services too, we also render some, let's say, Sanger sequencing. So maybe if you're students and then you want to do Sanger sequencing, you want to perform some NGS system as sequencing as well, we also render that services. And maybe you have your data and then you want some bioinformatics support, and also you want to do some animal genetic services, we render that services as well. So to talk more about our sequencing, we are the original partners with the PacBio, and when we compare the system to any other system available, this one accuracy is about 99.99%, and we have like smaller systems that can fit onto your desktop. We have the Onso and then we have the Review. So if you want more details about that, you can visit our booth, and then I would love to talk to you more on that. Okay, and aside that too, we have the mass array. We offer that services as well for your SNPs analysis. And when it comes to training, we also offer training as a way of promoting molecular techniques. So these are some of the pictures from the phylogenetics workshops we had in South Africa. And I think in Ghana, this one, we organized this in Noguchi, I think about 3 years ago. And in addition to, we also organize hands-on training. So maybe if you want to do DNA extraction, you want to learn how to do PCR, and you also want to do further analysis for with that, we offer that services in Nigeria, because currently the Ghana office, we are yet to set up our place for lab work. And when it comes to our partners, we've partnered with all these. So when it comes to your isolation kits, we've partnered with Zymo. So anything Zymo research, you can always come to Nkaba, and then I'll talk to you more on that. Currently, we have a promo for the RNA extractions. So if maybe you are having anything like that, we have 20% off for all our RNA extraction kits. And then when it comes to your PCR, we are the main, let's say, representatives when it comes to New England Biolabs. So from your, let's say, restriction enzymes for your PCR kits, if you are performing RT-qPCR, we have solutions from NEB. And also when it comes to equipment, we also represent Eppendorf. So currently we have a special Nkaba discounts for the Nexus. So if you're interested to know more about that, you can pass by our booth and I'll talk to you more about that. And then when it comes to instrument, we represent ICA. So for your centrifuges, your volters and all that, we represent that. So I think the new addition is that we've added some clinical side as well. So in the hospital settings, if maybe you are planning of setting up, let's say, a lab and then you need some analyzers and all that, we represent Siemens. And then for biorats too, we also provide their external controls. And when it comes to the molecular diagnosis kits, we've partnered, or during the heat of COVID, we've set up a lot of labs to test for viral loads, to test for COVID, and then to test for other stuff. And I know here in WACCBIP, a lot of students are using our Seegene kits, where they are using it for HPV and other stuff, which, if you need after-sales support, I've done it, and I'm also glad to welcome new students who will be interested in using our CG kits. And when it comes to COVID, the workflows is as represented. So I would say that in a nutshell, Nkaba is, let's say, we have position and a place where we provide after sales support even when you buy reagents from us. And also when it comes to our diagnosis kit like the CJ, if you want, let's say, explanation on how to set up your experiment, even how to analyze your data, that's my job. And I'm here to welcome new students in that field. Thank you very much.
Thank you very much. And just before I release you for lunch, one final notice to those queued for the Turbo Talks tomorrow. Make sure you submit your slides today, and one slide, right? And those who are queued today, I think 1 or 2 people missed it. You can join tomorrow's. All right. And I think we can go for lunch.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood. Okay.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients as we need a sample of her blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Doc, please, we are here.
Oh, good. I hope you are well. Don't worry. We are here to help you. All of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? We need a sign language interpreter.
Hey, stop looking at them.
Who told you I'm interested?
I can see it in your eyes. See.
Doris, they are deaf and dumb people.
It's true. Ah,
I hear these people are cares though.
how do you even introduce them as your friends?
Every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money?
Oh, I see. You are not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha.
Oh, boss.
Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
I think I can help.
Yes.
Okay.
Please.
Okay.
This is good. Then we get this for this.
Let me see how far.
You!
Hey! What is this?
Masha! Masha!
What is it?
Warn your son! Warn your son! Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son or come to my house?
Oh, Ejapia! What's wrong if Kofi plays with your son? Are we not from the same clan?
Hey! Don't you know there is a case on you and your husband?
Me and my husband?
Yes!
Me and my husband?
Yes!
Very good. My husband is in.
Did you say curse? All right. We'll see who that curse is on.
Deal with him! Curse him! Deal with him! But shipping, come on. Wow.
Jonathan, we can't find Amma.
Where is she?
I don't know, we've looked everywhere.
What? Yes. In that critical condition? Oh God. This is unbelievable. We have to find her. We have to find her at all costs. I'll handle it.
Thank you.
My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? Well, you know the people who did all the antenatal tests. How can you tell us our child is deaf? We'll solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. And your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. If that is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Chikere. Sumanga. Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghana cedis.
Your Highness, can we pay in cash?
Oh, the gods can take cash, even mobile money. Chikere, pala wenzu. Pala.
Azomagba balawezo wasumagba balawezo.
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali, Mali, Mali.
He said she was going to get better when we get home. But now her condition is worsening. Please help us, please.
This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I am going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. Jesus' name. Oh, Father. Jesus. Jesus' name.
Koyo, she's not getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong. We now have a better understanding of what genetics is and its implications on deafness.
How can we thank you, doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Oh, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, Doctor. Then I'll quickly arrange for the meeting.
Thank you. Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana wants to meet everybody at the village square. The great Asparrow has spoken. Ago! People of Asempa, Nana and the council request your presence. Nana wants to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son Kojo and daughter Agnes are here with some medical officers from Legon to educate us on the subject of deafness. and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Please, you have the floor now.
Thank you very much, Nana.
As part of our work as clinicians and researchers, we investigate what makes each person unique and different. We do this by examining your blood sample. How many of you here have ever given their blood for testing?
Blood. I can't give my family.
Blood.
My family. No. Blood is sacred. We don't give blood. Yes. It will never happen like that. I can't give my blood.
It is very important to give blood to be tested because every information that is used in building our body is packaged as genes, and this can be isolated from our cells in our blood sample. This we inherit from our parents. Please, can you bring the chart? You have to look on this chart. It will help you understand it better. Please.
Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribose nucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code. Genes variations are like spelling errors, where, for example, there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything the parents could have caused or prevented in any way. They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have 2 copies. One copy comes from the mother and the other copy from the father. In an autosomal recessive condition, an individual needs both copies of genes to be faulty for a condition to develop. The parents of a child usually carry one faulty gene of the gene pair. They are healthy carriers. It is estimated that every individual is a healthy carrier of several faulty genes, but a recessive condition only occurs when both individuals in a couple happen to carry the same faulty genes. When healthy carriers have children, there are 3 possible outcomes. 1. The child inherits both faulty gene copies and has the condition. 2. The child inherits only one faulty gene copy and is a healthy carrier like his or her parent. 3. The child inherits both working copies of the gene. So each time healthy carriers have children, there is a 1 in 4, 25% chance that their children could inherit both faulty gene copies and develop the condition. When an individual with a recessive condition have children, their children will usually all be healthy carriers of the condition. Their children could develop the condition if their partner happens to be a carrier of the faulty gene, but this could be a rare occurrence. Brothers and sisters of a healthy carrier could also be healthy carriers themselves.
So as you can see, features such as our height, complexion, color, including defects such as sickle cell and deafness, are all inherited from our parents. These traits are controlled by our genes. Have you noticed that every family has a feature that is passed on from generation to generation?
Yes. That is why our elders say the crab does not give birth to a bird. Yes. But I want to ask a question. Are you saying that deafness is not caused by curses or witches?
My elders and people of Asempa, that was what I believed until I could not find spiritual remedy for my daughter's deafness. That was why I gave in to genetic investigation.
I am glad this has come up. Please, from today, no one should associate deafness to witches or curses. As we have explained, every defective condition is as a result of our genetic inheritance. Although there are environmental factors.
People like Mr. Akoto and his family, and like my daughter, are all normal human beings. Their deafness can be explained through scientific investigation.
No, please, let us put it on record that with the advancement of science and technology, every medical condition can be scientifically examined, including deafness. So please, I will kindly entreat all of you to stop attributing deafness to spiritual causes.
Doctor, please. Some of our neighbors and the entire family are deaf. Does it mean that they spread it to one another? That is why we want Nana to send them out from our community.
No. Please, be rest assured. Deafness is not contagious.
Hey, thank God! Oh, my children.
Thank you. It's okay, woman. It's okay, woman. Please sit down. It's okay. Let them finish what they are saying.
Nana, all we need to do is to avail ourselves to be examined. It is possible some can be helped, but the most important thing is getting advice through genetic counseling and also having new babies screened for early detection. That's all we have to do.
Yo, my people, wisdom, they say, is like a baobab tree, and no one person can embrace it all alone. Today, we have been educated that the deaf people in our midst are our brothers and sisters. They are not under any curse. They are humans just like us. There is no curse on them. So we have to show them love and respect. And more importantly, we have to make ourselves available for genetic investigation.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, blood.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patient that's really the sample of her blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Doctors, you are here.
Oh, good. I hope you are well. Don't worry. We are here to help you. All of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? We need a sign language interpreter.
Hey, stop looking at them. Who told you I'm interested? I can see it in your eyes. See. Doris, they are deaf and dumb people. It's true. Ah, I hear these people are cursed though. How do you even introduce them as your friends?
Every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money.
Oh, I see. You are not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha.
Boss.
Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
Yes. I think I can help.
Yes.
Okay.
Please.
Okay.
This boy, then be cases for this.
Let me see how far.
I'll show you.
Hey! What is this?
Masha!
What is it?
Warn your son. Warn your son. Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son? Or come to my house?
Oh, Ejapia! What's wrong if Kofi plays with your son? Are we not from the same clan?
Hey! Don't you know there is a case on you and your husband?
Me and my husband?
Yes!
Me and my husband?
Yes!
Very good. My husband is in.
Did you say curse? All right. We'll see who that curse is on.
Deal with him! Chase him! Deal with him! But shipping, come on. Wow.
She has nothing to fear.
Doctor, we can't find Amma.
Where is she?
I don't know. We've looked everywhere.
What? Yes. In that critical condition? Oh God. This is unbelievable. We have to find her. We have to find her at all costs. I'll handle it. My friend, thank you very much for coming. I'll call you when she turns up.
shout.
I'll shout. What more tests do you need? Well, you know the people who did all the antenatal tests. How can you tell us our child is deaf? We will solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. And your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. If that is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Chikere. Sumangba. Baba, jamato, baba, sumangba, jamato. Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghana cedis.
Your Highness, can we pay in cash?
Oh, the gods can take cash. Even mobile money. Chikere, pala wenzu. Pala
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali.
He said she was going to get better when we get home. But now, her condition is worsening. Please help us, please.
This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I am going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. In Jesus' name. Oh, Father. Jesus' name.
Koyo, she's getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong.
See?
We now have a better understanding of what genetics is and its implications on deafness.
How can we thank you, doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Oh, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, doctor. Then I'll quickly arrange for the meeting.
Thank you. Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana want to meet everybody at the village square. The great Asparrow has spoken. Ago! People of Asempa, Nana and the council request your presence. Nana want to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son Kojo and daughter Agnes are here with some medical officers from Legon to educate us on the subject of deafness and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Please, you have the floor now.
Thank you very much, Nana.
As part of our work as clinicians and researchers, we investigate what makes each person unique and different. We do this by examining your blood sample. How many of you here have ever given their blood for testing?
Blood. I can't give my family.
Blood.
My family. No. Blood is sacred. We don't give blood. Yes. It will never happen like that. I can't give my blood.
It is very important to give blood to be tested because every information that is used in building our body is packaged as genes. And this can be isolated from our cells in our blood sample. This we inherit from our parents. Please, kindly bring the chart. You have to look on this chart. It will help you understand it better. Please.
Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribonucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code. Genes variations are like spelling errors, where, for example, there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything the parents could have caused or prevented in any way. They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have 2 copies. One copy comes from the mother and the other copy from the father. In an autosomal recessive condition, an individual needs both copies of genes to be faulty for a condition to develop. The parents of a child usually carry one faulty gene of the gene pair. They are healthy carriers. It is estimated that every individual is a healthy carrier of several faulty genes, but a recessive condition only occurs when both individuals in a couple happen to carry the same faulty genes. When healthy carriers have children, there are 3 possible outcomes. 1. The child inherits both faulty gene copies and has the condition. 2. The child inherits only one faulty gene copy and is a healthy carrier like his or her parent. 3. The child inherits both working copies of the gene. So each time healthy carriers have children, there is a 1 in 4, 25% chance that their children could inherit both faulty gene copies and develop the condition. When an individual with a recessive condition have children, their children will usually all be healthy carriers of the condition. Their children could develop the condition if their partner happens to be a carrier of the faulty gene, but this could be a rare occurrence. Brothers and sisters of a healthy carrier could also be healthy carriers themselves.
So as you can see, features such as our height, complexion, color, including defects such as sickle cell and deafness, are all inherited from our parents. These traits are controlled by our genes. Have you noticed that every family has a feature that is passed on from generation to generation?
Yes.
That is why our elders say the crab does not give birth to a bird. But I want to ask a question. Are you saying that deafness is not caused by curses or witches?
My elders and people of Asempa, that was what I believed until I could not find spiritual remedy for my daughter's deafness. That was why I gave in to genetic investigation.
I am glad this has come up. Please, from today, no one should associate deafness to witches or curses. As we have explained, every defective condition is as a result of our genetic inheritance. Although there are environmental factors.
People like Mr. Akoto and his family, and like my daughter, are all normal human beings. Their deafness can be explained through scientific investigation.
No, please, let us put it on record that with the advancement of science and technology, every medical condition can be scientifically examined, including deafness. So please, I will kindly entreat all of you to stop attributing deafness to spiritual causes.
Doctor, please. Some of our neighbors and the entire family are deaf. Does it mean that they spread it to one another? That is why we want Nana to send them out from our community.
No. Please, be rest assured. Deafness is not contagious.
Hey, thank God!
Thank you. It's okay, woman. It's okay, woman. Please sit down. It's okay. Let them finish what they are saying.
Nana, all we need to do is to avail ourselves to be examined. It is possible some can be helped, but the most important thing is getting advice through genetic counseling and also having new babies screened for early detection. That's all we have to do.
Yo, My people, wisdom, they say, is like a baobab tree, and no one person can embrace it all alone. Today, we have been educated that the deaf people in our midst are our brothers and sisters. They are not under any curse. They are humans just like us. There is no curse on them. So we have to show them love and respect. And more importantly, we have to make ourselves available for genetic investigation.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood. Okay.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patient that's really the sample of her blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Doc, please, we are here.
Oh, good. I hope you are well. Don't worry. We are here to help you. All of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? We need a sign language interpreter.
Stop looking at them. Who told you I'm interested? I can see it in your eyes. See. Doris, they are deaf and dumb people. It's true. I hear these people are kids though. How do you even introduce them as your friends?
You know, every physical condition has its medical cause.
It has nothing to do with curses.
Oh, hello, sir. Your money.
Oh, I see. You are not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha.
Of course.
Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
I think I can help.
Yes.
Okay.
Please.
Okay.
This boy, then be cases for this. Let me see how far.
You!
Hey! What is this?
Masha!
What is it?
Warn your son! Warn your son! Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son?
Oh.
Or come to my house?
Oh, Ejapia! What's wrong? If Kofi plays with your son, are we not from the same clan?
Hey! Don't you know there is a case on you and your husband?
Me and my husband?
Yes!
Me and my husband?
Yes!
Very good. My husband is in.
Did you say curse? All right. We'll see who that curse is on.
Deal with him! Chase him! Deal with him! But shipping, come on. What?
Doctor, we can't find Amma.
Where is she?
I don't know. We've looked everywhere.
What? Yes. In that critical condition? Oh God. This is unbelievable. We have to find her. We have to find her at all costs. I'll handle it.
Thank you.
My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? Well, you know the people who did all the antenatal tests. How can you tell us our child is deaf? We will solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. And your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. I think that is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Chikere. Sumagba. Bagba, jama sumagba, oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghana cedis.
Your Highness, can we pay in cash?
Oh, the gods can take cash, even mobile money. Chikere, pala wenzu. Pala. Azomagba, pala wenzu.
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali.
He said she was going to get better when we get home. But now, her condition is worsening. Please help us, please. This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I am going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. Jesus' name. Oh, Father. Jesus' name. Kojo, she's not getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong.
See?
We now have a better understanding of what genetics is and its implications on deafness.
How can we thank you, doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Oh, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, Doctor. Then I'll quickly arrange for the meeting.
Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana wants to meet everybody at the village square. The great Asparrow has spoken. Ago! People of Asempa, Nana and the council request your presence. Nana wants to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son Kojo and daughter Agnes are here with some medical officers from Legon to educate us on the subject of deafness and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Please, you have the floor now.
Thank you very much, Nana.
As part of our work as clinicians and researchers, we investigate what makes each person unique and different. We do this by examining your blood sample. How many of you here have ever given their blood for testing?
Blood. I can't give my family.
Blood.
My family. No, blood is sacred. We don't give blood. Yes. It will never happen like that. I can't give my blood.
It is very important to give blood to be tested because every information that is used in building our body is packaged as genes, and this can be isolated from our cells in our blood sample. This we inherit from our parents. Please, kindly bring the chart. You have to look on this chart. It will help you understand it better. Please. Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribonucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code. Genes variations are like spelling errors, where, for example, there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything a parent could have caused or prevented in any way. They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have 2 copies. One copy comes from the mother and the other copy from the father. In an autosomal recessive condition, an individual needs both copies of genes to be faulty for a condition to develop. The parents of a child usually carry one faulty gene of the gene pair. They are healthy carriers. It is estimated that every individual is a healthy carrier of several faulty genes, but a recessive condition only occurs when both individuals in a couple happen to carry the same faulty genes. When healthy carriers have children, there are 3 possible outcomes. 1. The child inherits both faulty gene copies and has the condition. 2. The child inherits only one faulty gene copy and is a healthy carrier like his or her parent. 3. The child inherits both working copies of the gene. So each time healthy carriers have children, there is a 1 in 4, 25% chance that their children could inherit both faulty gene copies and develop the condition. When an individual with a recessive condition have children, their children will usually all be healthy carriers of the condition. Their children could develop the condition if their partner happens to be a carrier of the faulty gene, but this could be a rare occurrence. Brothers and sisters of a healthy carrier could also be healthy carriers themselves.
So as you can see, features such as our height, complexion, color, including defects such as sickle cell and deafness, are all inherited from our parents. These traits are controlled by our genes. Have you noticed that every family has a feature that is passed on from generation to generation?
Yes. That is why our elders say the crab does not give birth to a bird. Yes. But I want to ask a question. Are you saying that deafness is not caused by curses or witches?
My elders and people of Asempa, that was what I believed until I could not find spiritual remedy for my daughter's deafness. That was why I gave in to genetic investigation.
I am glad this has come up. Please, from today, no one should associate deafness to witches or curses. As we have explained, every defective condition is as a result of our genetic inheritance. Although there are environmental factors.
People like Mr. Akoto and his family, and like my daughter, are all normal human beings. Their deafness can be explained through scientific investigation.
No, please, let us put it on record that with the advancement of science and technology, every medical condition can be scientifically examined, including deafness. So please, I will kindly entreat all of you to stop attributing deafness to spiritual causes.
Doctor, please. Some of our neighbors and the entire family are deaf. Does it mean that they spread it to one another? That is why we want Nana to send them out from our community.
No. Please, be rest assured. Deafness is not contagious.
Hey, thank God!
Thank you. It's okay, woman. It's okay, woman. Please sit down. It's okay. Let them finish what they are saying.
Nana, all we need to do is to avail ourselves to be examined. It is possible some can be helped, but the most important thing is getting advice through genetic counseling and also having new babies screened for early detection. That's all we have to do.
Yo, my people, wisdom, they say, is like a baobab tree, and no one person can embrace it all alone. Today, we have been educated that the deaf people in our midst are our brothers and sisters. They are not under any curse. They are humans just like us. There is no curse on them. So we have to show them love and respect. And more importantly, we have to make ourselves available for genetic investigation.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, blood. Okay.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients that's really the sample of her blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Doctor, you are here.
Oh, good. I hope you are well. Don't worry. We are here to help you, all of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? We need a sign language interpreter.
Hey, stop looking at them. Who told you I'm interested? I can see it in your eyes. See. Doris, they are deaf and dumb people. It's true. I hear these people are cursed though. How do you even introduce them as your friends?
Every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money.
Oh, I see. You're not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha.
Boss.
Oh, okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
Yes. I think I can help.
Yes.
Okay.
Please.
Okay.
This boy, then be cases for this.
Let me see how far.
I'll show you.
Hey! What is this?
Masha!
What is it?
Warn your son. Warn your son. Or else you will see what you do.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology. We make excellence the bar for training at WACCBIP.
We believe in excellence
in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and also that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department, and the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region, because each workshop we had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the ring. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Teresa, Osborne, Abida. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordon Awandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than $50 million US for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honour for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the dearth of genetics research in African people. And what was striking is it's a huge gap in knowledge. And Africans happen to be the most genetically diverse population. After my first postdoc, when I decided I wanted to move back to Ghana, I wanted to work in an area that had a huge impact. I mean, there are practically no genetic studies being conducted here. So I decided to focus on breast cancer, which happens to be the number one leading cancer in the world currently.
The center also focused on deepening its network with other African scientific centers.
Through my collaboration with WACCBIP, I think we've achieved enormous in our science. We've developed lots of talents that are actually beneficial to African institutions, which have also been exported across the world. I think WACCBIP is the next level of scientific institutions that we expect in Africa that do not only do research for publications, but also contribute to data that is used for translation for better public health across the great continents.
WACCBIP has trained more than 450 graduate students, masters and PhD students, as well as postdoctoral fellows from 19 African countries.
WACCBIP has more than prepared me, and I wouldn't say this because I've just stayed in WACCBIP, but for the fact that WACCBIP has given me the opportunity to go rub shoulders with my colleagues in other countries, and I'm willing to and I'm ready to take up any challenge anywhere.
The PhD training I had here was holistic, and indeed it has helped me gain that confidence and also to find solutions to problems.
The teaching environment at WACCBIP was quite unique. I found that management usually would invite visiting lecturers from either other institutions or outside the country to come in and teach us, and then teach us the practical ways of doing research and then thinking about problems to solve it through research. So
the fellowship that we were getting was about developing leaders in science, not just about training PhD students, but also building leaders for the next generation, or at least future scientists, future science leaders in Africa.
The opportunity to deepen the center's impact presented itself during the COVID-19 pandemic. WACCBIP had the right scientists and infrastructure in place to lead the science that informed the government of Ghana's policy response.
WACCBIP did play a very central role in our response.
We were able to have a strong impact and mobilize the teams to respond first for testing and then for doing the research that we're doing now.
One of the critical areas WACCBIP did excel was in the research, in the genomic sequencing and in the seroprevalence studies. And this really helped us not just to map out where the diseases are, but which variants we were dealing with, and especially when we started easing restriction.
During the COVID-19 pandemic, our researchers here, using the high-performance computing system, were able to assemble the genome of the virus, and as a result of that, gain a lot of insight into its transmission, subtypes, variation, and possible mutation.
Most of the work that is conducted at the center is at a molecular level. If you have a blunt cutlass, you don't think you can cut down a bigger tree. It's just the shrubs you can. But if you have a sharp cutlass, which is also basically the tools that we have, then we can confront bigger and higher molecular kind of questions and address them.
We have a tradition that we say in the center known as the WACCBIP machinery, and that comprises of a group of people who believe in a common goal and who are determined to make sure that they achieve excellence at everything that they do, whether it is research, whether it's training or teaching, and then the entire back end that nobody gets to see. Really teamwork under very diligent leadership that makes us achieve the excellence that we are known for.
I love working at WACCBIP because the leadership of WACCBIP identifies young talents and then give them opportunity to grow. I've been here 10 years because I believe that I've had opportunities that I wouldn't have had elsewhere.
We have created innovative and engaging ways of interacting with communities that we work with. We conduct and engage our communities in outreaches, cruise engagement activities, and a whole lot of activities that make science fun and exciting for the public.
WACCBIP has demonstrated that it is possible for us to remain in Africa and produce high-quality scientists.
I think its focus is on two things. One, doing the research, but also developing the researchers of the future. And that is where I think WACCBIP has a special strength, focusing on developing the researchers of the future.
We are training all these young people, but where are they going to work? If you look in the US or in Europe, majority of PhD graduates don't stay in academia. They go into industry, they go into a variety of jobs.
In Ghana and in fact most of West Africa, we don't have a biotech sector. We have some pharmaceutical companies, but most of them just do manufacturing. Because of that gap, many of the things that we've done have gotten to a dead end because the uptake is not there. And so we are trying to push for that next level.
We also need to build the ecosystem here of a biotech. So we have a program which is a biotech entrepreneurship program. And the idea is that now we are going to be seeding people who not just have the ideas, but they will try to do some startups. Of course,
the idea of building private sector-based research to complement the academic research that is being built at centers like WACCBIP, I think is a natural evolution of the development of the center. WACCBIP's goal is to train and seed science around the continent. What better way to seed it than through private companies that do the work like what we do here?
The next phase is on the shoulders of the next generation, the younger scientists that we brought here. Very talented group of people.
Happy 10 years, WACCBIP.
Happy 10 years, WACCBIP.
Happy 10th anniversary, WACCBIP.
Happy 10 years to WACCBIP.
Happy 10 years, WACCBIP.
happy, glorious and illustrious 10th anniversary, WACCBIP.
Happy 10th anniversary, WACCBIP.
many congratulations to everybody involved in the WACCBIP story.
Hello, how are you doing today? Oh no, I need a little sample of your blood for the lab. It's not painful, just a little sample of your blood. It's not painful, it's not painful. Blood. Okay.
Is everything okay?
No, please, there's a problem.
What is it?
I don't know how to communicate to the patients that we need a sample of her blood for the lab.
No. You see, that is why we need sign language interpreters in all health facilities. Don't worry, I'll handle it.
Okay.
Bring her to the lab.
Okay, please, I'll do that. Please, we are here.
Oh, good. I hope you are well. Don't worry. We are here to help you. All of us. To do that, we need a sample of your blood. A sample, Don't be afraid. Your blood, small sample, okay? For our diagnosis. You see, please, it's okay. Take it easy. Take it easy. Don't be afraid. Oh my God. How am I going to explain this to her? It's okay. It's okay. You take her back to the ward. You'll be fine, okay? We need a sign language interpreter.
At them. Who told you I'm interested? I can see it in your eyes. See. Doris, they are deaf and dumb people. It's true. I hear these people are friends though. How do you even introduce them as your friends?
You know, every physical condition has its medical cause. It has nothing to do with curses.
Oh, hello, sir. Your money.
Oh, I see. You are not deaf?
No, I'm not deaf.
That is interesting.
Yes.
Aha.
Oh,
okay. I will need your assistance to communicate with a deaf patient.
Oh, okay. Which hospital?
Legon Hospital.
Wow. Actually, I've just been transferred from the Alpha Clinic to the X-ray department.
Really?
Yes.
Wonderful. That makes my work easier.
I think I can help.
Yes.
Okay.
Please.
Okay.
This boy, then we guess this for this.
Let me see how far.
You!
Hey! What is this?
Masha! Masha!
Ah, what is it?
Warn your son! Warn your son! Or else you will see what you don't want to see and hear what you don't want to hear. How many times have I not warned him not to play with my son? Or come to my house?
Oh, Ejapia! Ejapia! Ah! What's wrong if Kofi plays with your son? Are we not from the same clan?
Hey! Don't you know there is a case on you and your husband?
Me and my husband?
Yes!
Me and my husband?
Yes!
Very good. My husband is in.
Did you say case? All right. We'll see who that case is on.
Deal with him! Chase him! Deal with him! But Chippy, come on. Wow.
Doctor,
we can't find Amma.
Where is she?
I don't know, we've looked everywhere.
What? Yes. In that critical condition? Oh God. This is unbelievable. We have to find her. We have to find her at all costs. I'll handle it.
Thank you.
My friend, thank you very much for coming. I'll call you when she turns up.
I'll shout. What more tests do you need? We know the people who did all the antenatal tests. How can you tell us our child is deaf? We will solve our own problem. Let's go.
Excuse me, sir. Is there any problem?
What I say is final. We are not going to any pastor. We are going straight to the village.
I am not ready to take my daughter to the shrine.
No one in my family is deaf. And your family too, there is none. So how can you tell me my daughter is deaf? We must look into it. This is purely spiritual.
What about going to do the test? So the doctor can determine how to help her.
It is my daughter who is deaf. What do they need my blood for?
Our blood. If that is what will help her.
Blood is sacred. I am not giving my blood to any doctor. I will call Papa at the village to arrange a meeting with the diviner.
Thank you.
Chikere. Sumangba. Baba, jamato, baba, sumangba, jamato. Oh, great diviner, eye of the gods, the great owl whose gaze pierces through the thickest shadow and the darkest night. There is a deity that must be appeased in your family. She has been abandoned and has come with a vengeance of deafness. You must pay the due and purge your child. 3 rams and 3,000 Ghana cedis.
Your Highness, can we pay in cash?
Oh, the gods can take cash, even mobile money. Chicken, pala wenzu. Pala.
Hey! Something is happening to her.
Don't worry, my daughter. The spirit is leaving her. Mali.
He said she was going to get better when we get home. But now her condition is worsening. Please help us, please. This is highly demonic. She needs deliverance. I can feel it in me that there's a bad spirit inside this child.
Prophetess, can you deliver her?
That is exactly what I am going to do. Let us bow before the Lord. Jesus, we trample on every serpent and scorpion. According to the word, we rebuke every demonic power of high places, every altar that stands against the authorities of Jehovah. Be brought low. Jesus' mighty name. Jesus' name. Jesus' name. Oh, Father. Jesus' name. Koyo, she's not getting better.
What should we do now?
Let's go back and see the doctor.
Okay.
Let's go.
Doctor, thank you very much for the explanation. We got it all wrong.
See?
We now have a better understanding of what genetics is and its implications on deafness.
How can we thank you, doctor? Many people out there are not privy to this information.
Especially the people of our village. Doctor, there is a deaf community in our village. Is it possible for you and your team to come and explain this to the people?
Oh, that's a very good idea. It's an opportunity to reach out to the community and educate them. We will. Why not?
Thank you very much, Doctor. Then I'll quickly arrange for the meeting.
Thank you. Thank you. Thank you. She's as normal as any other child. Just take good care of her. Okay? It's okay. Don't need to cry.
Ago! Nana and the council request tomorrow morning. Nana want to meet everybody at the village square. The great Asparrow has spoken. Ago! People of Asempa, Nana and the council request your presence. Nana wants to meet everybody at the village square. The great Asparrow has spoken.
Nana and Council welcome you all to this urgent but very important meeting. Nana want to cut straight to the point. Nana, speak.
People of Asempa, the issue confronting us is a very serious one which needs to be addressed. I'm not an expert myself, but our forebears have told us over the decades the mysteries surrounding deafness in our community. Much as we have tried, we've not been able to find answers to this problem. But today, our son Kojo and daughter Agnes are here with some medical officers from Legon to educate us on the subject of deafness and its possible causes. Dr. Wo and Dr. James, you are most welcome to this community.
Thank you very much, Nana.
Please, you have the floor now.
Thank you very much, Nana.
As part of our work as clinicians and researchers, we investigate what makes each person unique and different. We do this by examining your blood sample. How many of you here have ever given their blood for testing?
Blood. I can't give my family.
Blood.
My family. No. Blood is sacred. We don't give blood. Yes. It will never happen like that. I can't give my blood.
It is very important to give blood to be tested because every information that is used in building our body is packaged as genes, and this can be isolated from our cells in our blood sample. This we inherit from our parents. Please, kindly bring the chart. You have to look on this chart. It will help you understand it better. Please. Our bodies are made up of billions of cells. Inside each cell is our genetic material. Chromosomes are the packaging of our genetic material. We usually have 23 pairs of chromosomes, and we inherit one copy from a mother and one from a father. If we unwind the chromosomes, we can identify the genes. Genes are the basic building blocks that makes proteins. Genes are like the blueprints for making a machine, and proteins are the individual components of the machine. Proteins makes us who we are and help us grow and function properly. Our genetic materials is made up of our DNA. Deoxyribose nucleic acid is the very long chain of 4 chemical letters: adenine, guanine, thymine, and cytosine that combine to make our genetic code. We all have some variations in our genetic code. Genes variations are like spelling errors, where, for example, there's a T instead of a C. A variation in the gene that makes it faulty or not working properly is called mutation. Faulty genes are caused commonly simply through chance. They have to occur in one individual for the first time at some stage. They are not related to anything a parent could have caused or prevented in any way. They occur because the process that replicates and divides their genes so we can pass it on to the next generation is complex, and mistakes occur. For each gene, we have 2 copies. One copy comes from the mother and the other copy from the father. In an autosomal recessive condition, an individual needs both copies of genes to be faulty for a condition to develop. The parents of a child usually carry one faulty gene of the gene pair. They are healthy carriers. It is estimated that every individual is a healthy carrier of several faulty genes, but a recessive condition only occurs when both individuals in a couple happen to carry the same faulty genes. When healthy carriers have children, there are 3 possible outcomes. 1. The child inherits both faulty gene copies and has the condition. 2. The child inherits only one faulty gene copy and is a healthy carrier like his or her parent. 3. The child inherits both working copies of the gene. So each time healthy carriers have children, there is a 1 in 4, 25% chance that their children could inherit both faulty gene copies and develop the condition. When an individual with a recessive condition have children, their children will usually all be healthy carriers of the condition. Their children could develop the condition if their partner happens to be a carrier of the faulty gene, but this could be a rare occurrence. Brothers and sisters of a healthy carrier could also be healthy carriers themselves.
So as you can see, features such as our height, complexion, color, including defects such as sickle cell and deafness, are all inherited from our parents. These traits are controlled by our genes. Have you noticed that every family has a feature that is passed on from generation to generation?
Yes. That is why our elders say the crab does not give birth to a bird. Yes. But I want to ask a question. Are you saying that deafness is not caused by curses or witches?
My elders and people of Asempa, that was what I believed until I could not find spiritual remedy for my daughter's deafness. That was why I gave in to genetic investigation.
I am glad this has come up. Please, from today, no one should associate deafness to witches or curses. As we have explained, every defective condition is as a result of our genetic inheritance. Although there are environmental factors.
People like Mr. Akoto and his family, and like my daughter, are all normal human beings. Their deafness can be explained through scientific investigation.
No, please, let us put it on record that with the advancement of science and technology, every medical condition can be scientifically examined, including deafness. So please, I will kindly entreat all of you to stop attributing deafness to spiritual causes.
Doctor, please. Some of our neighbors and the entire family are deaf. Does it mean that they spread it to one another? That is why we want Nana to send them out from our community.
No. Please, be rest assured. Deafness is not contagious.
Hey, thank God! Thank you. It's okay, woman. It's okay, woman. Please sit down. It's okay. Let them finish what they are saying.
Nana, all we need to do is to avail ourselves to be examined. It is possible some can be helped, but the most important thing is getting advice through genetic counseling and also having new babies screened for early detection. That's all we have to do.
Yo, my people, wisdom, they say, is like a baobab tree, and no one person can embrace it all alone. Today, we have been educated that the deaf people in our midst are our brothers and sisters. They are not under any curse. They are humans just like us. There is no curse on them. So we have to show them love and respect. And more importantly, we have to make ourselves available for genetic investigation.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space, and
my name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meeting, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to be working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
Grant from Carroll Moore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation. The snake venom talk that has just happened was interesting because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas.
Okay,
and I thought that was really well done.
Just about getting ready to start the afternoon session.
10 years.
So just like yesterday, if you know somebody outside, please text them,
elsewhere in Africa,
call them, tell them we're
far.
a little behind schedule. So we'll be starting as soon as we have the room 3/4 full.
And will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's GISA, South Africa. And our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center, and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa. And it's made me want to fight harder with the South African government to allow us to get visas for students, so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
WACCBIP came out of the energy of many young people in the department.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately, I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here that being presented. I also think it's important to have people from around the world coming to this meal, and so all of these have impressed me over this week. I've also had really great interactions with trainees, and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway, at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to be working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between the London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that it is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
Grant from Caramore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. Yeah. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So.
All right. Good afternoon, everyone. I hope everyone had a very nice lunch and enjoyed the deliberations at the poster sessions. As we all know, there's never enough time for posters, and I hope we'll build on this so that tomorrow we eat quickly and then spend more time at the posters so that we can finish and be back in time. This is what I always consider to be one of the highlights of the conference, which is a panel discussion to really talk about putting things in granularity. And the thrust of this panel today is basically to look at how basic science research, particularly emerging from WACCBIP and the University of Ghana, feeds into the public health implementation pipeline, vaccine development policy, and biotech innovation. And we have 3 very well-placed individuals and experts to join me to discuss this in very succinct and hopefully tangible terms. So I will invite them to join me up on the panel, and we'll begin what I hope will be quite an engaging discussion. So I'll call Dr. Faisal Nuhu, who is the Deputy CEO at the National Vaccine Institute. Dr. Kezia Malm, who is the former program manager at the National Malaria Elimination Program. And Dr. Yaube Diako, who is first a WACCBIP fellow and more importantly, the CEO of Yamachi Biotech Incorporated. Okay. Good afternoon. So, I think before we start, I'd like all of you to introduce yourselves, starting with Dr. Nuhu. Yes, starting with Dr. Nuhu. Talk about your role at the National Vaccine Institute and how the National Vaccine Institute, how you sit and how you interact with us as academia to drive the public health.
Thank you very much, and it's a privilege to be here. Yesterday when I mounted here, I said your team, Evolving African Science from Capacity Building to Discovery and Impact, was as if you have come to National Vaccine Institute and said, what sort of team would you guys want us to put here? It speaks really to the core of what National Vaccine Institute is all about. I am the Deputy CEO, and I know yesterday I mentioned my boss was supposed to be here. He was called to equally important venture, so he's asked me to step in. NVI, in short, is the Ghana government and the people of Ghana's response to what I call vaccine racism following the COVID-19. People would like to call it vaccine nationalism. But I see, we all saw during COVID-19 that the Global South literally were left to their fate. And Ghana, the Ghana government at the time and the people of Ghana decided that no more would the life or the health of our citizens be determined by other capitals other than Accra. We will determine our own fate in Accra. So a committee was set, and that committee was led by scientists. You know, it goes to say, when times are hard, we have only one trusted partner to turn to, scientists, not politicians. And then the scientists deliberated and gave rise to National Vaccine Institute to oversee institutional arrangement that will lead to vaccine research and development as well as vaccine manufacturing. And once those institutional arrangements have been made, then National Vaccine Institute is also mandated by law to supervise vaccine R&D as well as vaccine research in the country. So that is what NVI is all about. And I'm the Deputy CEO. Thank you very much.
Thank you very much. So to Dr. Malm, I'll ask the same introductory question.
Thank you very much. It's a pleasure to be here. Good evening. So I am Kezia Malm. I just stepped down as the program manager for the National Malaria Program. And I believe I'm here because of that role and not the new role that I'm yet to experience. But basically, The National Malaria Program is the institution under Ghana Health Service that is responsible for coordinating all activities when it comes to malaria. So working with all partners to ensure that malaria is no more a disease of public health significance. And as a program, we do work with researchers quite